All right, so welcome to the RAES Regenerative Aesthetics Exosome Summit 2023, hosted by ExoCoBio and Benev. My name is Diego and I'll be your host tonight. I would like to welcome first Ethan Min. He's the CEO of Benev. Please welcome him.
It's kind of dark. Let the light be. Yes, great. Hi, everyone. My name is Ethan Min. I'd like to start it off of recognizing the people who put this event together. ExoCoBio. There are so many people here from ExoCoBio that actually put so much effort, including Mr. Joe, Ms. Dohi, and I'd like to give a big round of applause to the ExoCoBio team. Please. And there are about 26 countries being represented in this room right now. And that's amazing to us. Last year, I believe we have about 18 countries. Huge shout out to all the distributors who are here, all the doctors that came all the way to Korea. Thank you so much for being out here. Let's give us a round of applause to get to. I'd like to briefly share my personal, or Benev's personal journeys along with ExoCoBio came all the way to be here right now. I'm standing in front of you. Exosome is the big topic. Small particle, but it's the next big thing. And that's our slogan for US market for the last 5 years. It's a nanovesicle that contains microRNAs, growth factors, lipid-generating enzymes that are very, very effective for a skin rejuvenations, anti-inflammations, and so on and so forth. And then you guys probably heard enough of it. And then again, today, a lot of speakers are going to talk about that from the science aspect all the way to the clinical cases. But I just wanted to kind of draw your attention to the story where the particle size of the exosome is very similar to virus. And we just have experienced a global pandemic and how this tiny virus could be deadly and could be powerful forces to create a lot of damages on our humanity, right? And all of us have a lesson or two from this pandemic, and we overcame, and we're here. For me, there are significant lessons that I learned, and that are very powerful from this pandemic of COVID-19. I'm glad that I'm here standing in front of you guys, obviously. There's a reason why. I'd like to introduce and actually get your attentions to the book called Tipping Point by, I'm sure you guys heard of the, Malcolm Gladwell, right? He talks about a tipping point, and that is a magic moment when idea or trend, social behavior crosses a threshold, tips, and spread like wildfire. And that's the definition he made. And he goes on and basically talks about how he compares the concept of tipping points for pandemic, and also that can happen in businesses and also social phenomenon or movement. That he said that in order to create one contagious movement, you often have to create many small movements first. And that's the part that I'd like to share today that led me, myself, to all the way to be where we are with ExoCoBio. That since 2020, Benev, after we kind of coming out of the pandemic, joined force together with ExoCoBio and started this small contagious movement. And we did that by pioneering and paving the way for introducing and spreading this exosome concept into the medical aesthetic market. And we wanted to focus on regenerative aesthetics when we were actually starting doing so. Attending 80 different congresses every year between two companies, when there was no concept or no one was talking about it. Bringing the speakers to all these places, to sharing what we know then and impact and then the benefit of that. doing our own series of summits, including RAES starting from last year's, and having distributors all around the countries and to spread these good news. And that was obviously something that we started off, but if it wasn't for all of you guys, it probably was impossible, right? And Gladwell points out in his book and talks and quote, if you want to bring a fundamental change in people's belief and behavior, you need to create a community around them where those new beliefs can be practiced and expressed and nurtured. Today at RAES and this Regenerative Aesthetic Exosome Summit, we are here as a community of believer on exosomes. And I'd like to highlight all of you guys that we are at a tipping point. We're living in the tipping points of spreading this good news of exosome. Instead of fear of living in a pandemic of the virus, we are living in a joy of good news, right? That are spreading fast. The exosome is next big thing in the medical aesthetics. And 2018 is when we first started off. And in US, we have 3,500 accounts alone that are carrying out using our products. Globally, we have 15,000 providers around the globe already using our products. And there were already about 1 million cases of the treatment utilizing our products globally. And this is not about to stop. It is growing faster than ever. We definitely are in tipping point. Today, I have to basically call out the guy who's responsible for this. And I know all of you guys probably know this name, Mr. Jo Hyung-sung. And he is really the person who's responsible for this tipping point. Mr. Jo, would you please come out and stand for the crowd? Get the standing ovation. Let's give him a big round of applause.
Founder and CEO and CTO of ExoCoBio. First of all, it's our privilege to have our great minds in global exosome industry. We are very proud to present our today special scientists and doctors and distributors at the same time, at the same place. I believe, so it was my great fortune to start ExoCoBio about 7 years ago. And actually, just after incorporation, I went to Missionville, California to see Ethan, and we found that the two guys had the same idea and strategy and vision on action-based regenerative aesthetics. Also, before I incorporate ExoCoBio, I tried to co-work with one great scientist, Dr. Sai-Kyung Lin. She's here, and she is the world's first MSC exosome discoverer. Next slide. Okay, please let me just share 7 or 8 slides for my presentation. You can check the Google trend on how many people are trying to find exosomes. As you can see here, for the last 20 years, there has been very steady and nice increase in terms of search. When I start my presentation, I usually start with this slide with 9 questions. From my experience and my journey for the last 7 years, I found that these 9 questions are very important to, especially for you to decide or find or how to move forward in terms of in this exosome-based industry. Why ExoCoBio has been committed to be the leader now and in the future too. And why we are the leader? Because we are the biggest investor in exosome industry. ExoCoBio was strongly backed by Korean venture capital investors, and we have about 10 investors back there, and they supported from the beginning ExoCoBio to be the global leader. So that's why we have a brand new and top tier manufacturing facility named ExoGMP. I believe that most of you guys visited our manufacturing facility to check the future of exosome-based drug development. And in my view, the most important thing is publications or scientific evidences. So we spent a lot of money to prove the feasibility and the potency and the safety of exosomes with our 16 publications. Especially this year, we have generated 6 publications. 3 of them are non-clinical, and the rest of them are clinical studies. And actually, Dr. Jan Rattebar here, he's one of the top exosome scientists. He has been guiding us. He's managing our R&D team as our chief strategy officer. And then to protect our business, our providers, our distributors, and our partners around the world, we have generated more than 55 patents. We are in terms, definitely we are the leader to find a new scientific discovery and training patents. And also for last 4 years, we have been seeing about 1 million cases around the world, which means that our technology and our product has been proved in terms of efficacy and safety. So please remember these 9 questions and to choose your right technology. Here, this is yesterday. This year, I invited a very special guest. You can see that Kevin Hickok. He's a CTO of Directed Biologics, which is another US exosome company. And he's the only person who has phase 3 clinical study based on MSG exosome to treat COVID-19 ARDS. So he's an expert in exosome CMC, and ExoCoBio hope to co-work with him. And in the middle, as you can see, there is Dr. Sai-Kyung Lin, and she has been always with us. Actually, she has been scientific advisor for the last 6 years. Again, Dr. Jan Rattebar, he discovered for the first time RNA delivery mediated by exosomes. And also in Seoul, we have about 110 people for R&D, manufacturing, quality, something like that. And another about 40 people in Mission Viejo, California. They are all committed. And you saw this, the world's largest GMP manufacturing facility for exosome production. And we have been investing in this facility $26 million. And we hope to provide more products. And our plan is to provide 4.5 million vials per year, which is about $200 million worth from next year. And in this building, you are seeing that the actual operation to produce fully GMP compliant exosomes. And of course, we need to upgrade this process further, but I believe that the production capacity and the quality control level is the top tier. We are second to none. Next slide, please. So we call our manufacturing process SSCRT. It's patented in Korea. But our major pipelines are human stem cell derived exosome and plant stem cell derived exosome. We do not use the whole plant. We are utilizing plant stem cell derived exosome for best quality. But we have 2 different types of exosomes, from microbes Or we have a very nice publication this year on exosomes derived from animal or canine mesenchymal stem cells too. And most of our exosomes have been registered as cosmetic ingredient for now, but definitely in the future we hope to file an IND in the future, maybe 2 years. Next slide. So this is the part of our scientific data. You can see that for next generation regenerative aesthetics, we found 3 pillars of mechanism of action. The first one is anti-aging, the second is rejuvenation, the third is anti-inflammation. I know, but I'm talking pillar business, but our skin is the biggest part of our organ, which contains the biggest number of stem cells. So in our view, the anti-aging of our skin starts from the loss of adipose stem cells and the loss of their exosomes. So by providing high quality exosomes, we hope to be the ultimate next generation regenerative aesthetics. And part of our commitment is to develop RAES stem cell exosomes. We found, actually, under the guidance of Dr. Jan Rattebar, we just submitted our manuscript for publication on RAES stem cell exosome. Actually, this is the world's first plant stem cell-based exosomes. And we found that some of the science or scientific discoveries they are very similar to humans. But one thing is that what we found is that RAES stem cell EVs can induce our cells, our skin cells or stem cells to release more exosomes. So I believe we found that there is a strong link between plant stem cell derived exosomes and our skin, maybe. And as I mentioned, that our doctors or our providers are generating more and more clinical cases around the world to help patients. We found a lot of cases, and this event, you will see about 10 very nice cases generated by our providers. Next. That's why we have this award. As you know, ExoCoBio, our product became the best cosmetics this year, AMW Monaco. And as far as I know, ExoCoBio is the first Asian company to achieve this trophy at AMW Monaco. We are very proud to this event. Next slide. So again, we really appreciate your joining this event. We have 200 participants from 26 countries. Thank you so much. Next slide.
Thank you. So now we would like to welcome Dr. Michael Gold. We're about to start the first session of the night. Please get comfortable. We have 5 hours of a lot of science and cases, so please welcome Dr. Michael Gold.
Hi, everybody. So first I want to, from all of us here, we want to thank Byung and Ethan and all the teams from ExoCoBio and Benev for putting on this event and bringing us all here, and beautiful Seoul, and thank you all for being here, and how about a big hand for those two? So I'll tell you a little bit about me later, but I'm going to be the chair of this first session, and I'm going to use a little cheat sheet because our first speaker this is a privilege, right? You're all in for a privilege. So Dr. Jan Rattebar is a Swedish clinical allergist, scientist working on transitional research, and he's the former director of the Keftering Research Center in the University of Gothenburg. For our world, he has contributed to the early research of extracellular vesicles, such as exosomes and microvesicles as shuttles of RNA molecules between cells. And they were the group that showed mRNA in exosomes are functional when taken up in recipient cells. He is also the part of the European Academy, a member of the European Academy of Allergy and Clinical Immunology, but he started and was the first president of the International Society for Extracellular Vesicles, which is a society we all need to be part of, okay? So you can tell us more about that. And again, he is one of the 3 reasons that I'm putting out here why we are here today with our other 2 distinguished guests. Thank you all for being here. So John, you are up first, and your topic is harnessing exosomes for advanced biologic therapies.
Thank you so much. It's an honor to be here, actually. I've been working on exosomes or extracellular vesicles, and I will jump between those 2 different wordings, terminologies. I hope you forgive me for that. I've been working on it for almost 20 years. And just after we published our first paper, people wondered whether we were crazy because what are those exosomes? Nobody knew what the exosomes were, and it took a while for people to really pick up, and actually almost 10 years before this first company started. And it's a privilege for me actually to be here, because this community is the first to adopt the exosomes for clinical use in a professional way. So I think it's great. Now I'm going to talk a little bit generally about extracellular vesicles and what they do. I come from University of Gothenburg. I'm involved in International Society for Extracellular Vesicles. I'm the editor-in-chief for the journal, which has an impact factor of 17. I consult for ExoCoBio. I've done so for almost 3 years. I have my own startup company in Gothenburg in Sweden. Now, exosomes, I'm sure many of you just heard about exosomes a few years ago and wonder what are they. And actually, when we start thinking about what they are everywhere. They are everywhere in our body. And here's a bronchial biopsy from a young man actually, healthy man, where you can see fibroblast here, myofibroblast perhaps, but you can see a lot of vesiculation around this cell. But you can also see round structures quite far away from the cell. And here is another magnification where you can see down to 50, this is 50 nanometers, this little thing, which is the smallest size of an extracellular vesicle. So our whole body and interstitial fluid has these exosomes in it. And maybe the interstitial fluid is not a fluid, but actually colloid because of the sometimes high concentration of extracellular vesicles. So all the cells in the body release extracellular vesicles. All the body fluids that we have different amounts of extracellular vesicles in them. And it varies from body fluid to body fluid. And we exchange these vesicles between us in kissing, for example, or lovemaking or things like that. There's a huge amount of exchange of extracellular vesicles between 2 individuals. If you look at these vesicles, they are double membrane vesicles that have a lot of membrane proteins on the surface. So these are the standard proteins, the CD81, 63, and CD9, but also many other proteins. And you can engineer these vesicles to have certain proteins that enhance their traffic to specific cells. And we've been working a little bit on that in my lab over the last few years. And as you mentioned and Byung mentioned, I would say, stumbled on the extracellular RNA in exosomes 2004 or 2005, and then we wondered what was that, and then we figured out that RNA is certainly not staying within the cell, which was a dogma at the time, but actually leaves the cells and can be taken up by another cell and can be functional in the recipient cell. So that was a pretty big thing at the time. Of course, I lost grant because that was an artifact most likely. Now it's going much better, fortunately. So I'm a clinician background, so I want to utilize this system to develop medicines and use extracellular vesicles as medicines and to deliver things, but also to use their intrinsic capacity. And you have to be very careful of choosing the right source. So we will hear more about mesenchymal stem cell exosomes, a little bit from myself and also from Sai-Kyung Lim a little bit later today. I will show you a little bit of data, just one slide of data with bacterial vesicles, which actually are very powerful as well. or you can use so-called engineered cells, which are pretty much inactive. The exosomes are not regenerative in their own right, but they can be engineered to carry molecules that are therapeutic or regenerative or anti-inflammatory. And that's probably future development we'll see in the field. So just one slide on bacterial vesicles. And we developed a specific bacterial vesicle from E. coli that became non-toxic. So bacteria release vesicles all the time, and those vesicles contain LPS and a lot of other molecules that cause a lot of inflammation. Actually, they can cause sepsis just by themselves. And we used these vesicles. We detoxified these vesicles, so they were not causing those side effects. And then we've tested them in different systems. We have created vaccines with them, so we have bacterial vaccines using these vesicles. But we also tested them in cancer. So we inject these vesicles directly into tumor tissue, and we tested multiple types of tumors, and most of them it works. And this is a rapid-growing melanoma tumor that doesn't inhibit their growth with checkpoint inhibition. But with 2 different doses here of bacterial vesicles, we can actually totally stop the growth of these tumors in these mice. And we've done a lot of research, probably 4 years of R&D with this approach, and our goal now is to, within the next 2 years, try to go into clinic with these bacterial vesicles. But the absolute opposite then is to create anti-inflammatory treatment, not to induce immunity, but actually inhibit inflammation. And clearly the mesenchymal stem cell exosomes are a preferred platform for that, because they have this intrinsic anti-inflammatory activity. And you want that if you want to treat inflammation, and maybe you can enhance them by supercharging them, by loading them with different types of molecules. And then I mentioned the HEC293 cells that by themselves are not regenerative, but can be engineered, and that's a big part of our research right now. So we should be aware that the target cells, most of the target cells are immune cells in the body. Also in the cancer, for example, that I just showed, dendritic cells take up the bacterial vesicles and process them and create immunity against a tumor. But also macrophages are responsive. So the mesenchymal stem cells can be taken up by these 2 major regulators of inflammation or immunity and attenuate that reaction. And that's an important part of what the mesenchymal stem cell exosomes can do. Now, one thing I want to remind people, and not many people know this, right? But stem cells are not one thing, not at all. So there's a lot of hype around inducible pluripotent stem cells. And here are exosomes that we purchased, actually. We purchased the supernatant and isolated vesicles from ourselves from iPSCs. And you can see here that these vesicles are not anti-inflammatory at all. They are pro-inflammatory. So there are companies trying to develop these iPSC exosomes as anti-inflammatory treatment, but I would be questioning that. Whereas the mesenchymal stem cell derived vesicles are anti-inflammatory. And in normal doses, you get around 50% inhibition. This is a maximal dose, I would say. Now, the first study I was involved in with mesenchymal stem exosomes, which made me really excited about this, was a study, collaboration with a group in Southern California, where they studied a model of MS in the mouse. So this is a neurodegenerative model. And this mouse right here was treated with exosomes and had the induction, and this one was not treated with that. So you can see quite pronounced. Actually, this is only single dose treatment as well. So it's pretty spectacular effect that this inflammation or this inflammatory process was attenuated in this neurodegeneration. And here are 2 types of exosomes that were given, and both of them significantly reduced this MS-type inflammation. So if you look at the literature of mesenchymal stem cell exosomes, there are a lot of basic data. And I would say basically whichever animal model of inflammation that you put mesenchymal stem cells into, you will see an anti-inflammatory effect. So there is clearly an intrinsic anti-inflammatory effect of the mesenchymal stem cell exosomes. So there are not too many clinical trials, unfortunately. I will not go into the split phase study or the studies that ExoCoBio have done. Others will do that. But there are a few that have come out in the last few years, and some of them have focused on COVID and cytokine storm in COVID, and have indicated safety and maybe some anti-inflammatory effect, but the studies have been small and not double-blind and placebo-controlled. This is from Iran, and this is from China. Another approach that you might have heard of using exosomes to treat COVID cytokine storm is this engineering of CD24 on the surface of the exosomes. And that creates an intrinsically active anti-inflammatory exosomes. Now that's from HEC293 cells. And this Israeli group, they tested these vesicles with CD24 in COVID in 2020 and published in 21. And they did have a significant effect in these patients. But again, the study was not controlled. So as I said, most of the maximal effect is mostly around 50% with extracellular vesicles from mesenchymal stem cells. And this is in vivo experiments of sepsis in our lab. And we consistently see around 50% inhibition of inflammation. Now, as a clinician, that's not sufficient for us. We want more than 50% inhibition of a severe inflammation, certainly. So, if we can move forward, if the mouse could work, we have developed a method by which we can create vesicles directly from the cells, which we call synthetic eukaryotic vesicles, and we empty these vesicles. They don't have DNA and RNA, they're kind of empty, and that allows us to load them with different things. And you can see here that this effect of the exosomes, here are the exosomes, here are the empty synthetic vesicles, it's exactly the same, so you don't reduce the effect at all. This looks complicated. It's really not so complicated because we're targeting this is a cascade of inflammation that happens, for example, in sepsis, and you have activation of TLRs, toll-like receptors, and there are a whole number of different second messengers that are activated to create this severe inflammation. We have targeted with a peptide a GRAIL target, which is MyD88, which is impossible to target with small molecules. So when we add this peptide in vitro, we see some anti-inflammatory effect. We see the same level of anti-inflammatory effect with the synthetic vesicles, but we combine them and get these peptides inside of the vesicles. We have around 85% inhibition of inflammation. In vivo, the peptide is totally inactive. It doesn't get to the target cells, whereas the combination here is again about 85%. So you need to get these peptides inside of the vesicles, which allows them to be taken up by a recipient cell, and there the peptide targets the MyD88 and stops that severe inflammation. So you can imagine using this in the emergency ward in a patient that has a severe inflammatory situation. So in summary then, mesenchymal stem cells are intrinsically biologically active. The efficacy to a great degree is some activities is present in the stuff that is inside the microRNAs and so on, but a lot of the effect is actually located in the membrane because you can remove a lot of the cargo and still have effect. So we've loaded MyD88 peptide, and we've loaded cyclosporin A and some other molecules, and we always see synergy, so you can enhance that anti-inflammatory effect of the mesenchymal stem cell exosomes to quite pronounced effect. Now, of course, when you are treating patients, you need to have a GMP manufacturing. That's very important because you need to concentrate, you need to purify, you need to characterize the vesicles in a good way. And that's very important for quality control. And of course, any medicine that we use this GMP manufacturing approach. We need to determine which proteins are there, which impurities are there, and which proteins that should be there. Here are some examples that have been discussed. One thought among some people is that we could isolate exosomes from blood and use them as therapeutics. Now we should remember that when you isolate exosomes from blood using normal techniques such as centrifugation and so on, you get a huge amount of lipoproteins. And here you can see particles that are isolated from blood. And they don't look anything like the exosomes. This is actually highly purified exosomes through multiple steps from blood that have this cup shape, donut shape, whereas the rest of the blood is extremely contaminated with lipoproteins. So be aware of that. In summary, I think there's a great opportunity of developing mesenchymal stem cell exosomes as therapeutic. We can make them really powerful if we develop techniques to load those vesicles with appropriate molecules. The MyD88 peptide is only one example. There are other opportunities as well. Cyclosporin A also only one of several opportunities, but there's certainly great chances of developing exosomes. If we look a little bit later into the future, I think the HEC293 exosomes, maybe 3 or 5 years from now, will be something that we will be talking about much more. Thank you very much for your attention.
All right, that was fabulous, and it was a great introduction to what we're here for. And again, I think for a lot of us, having these folks here is very important for us because, again, for a lot of us, this is brand new. And we hope that when you're done this afternoon, that the brand new becomes something that really makes sense and is really important. So I'm going to talk about, I'm going the wrong way. Wait, you want to get me back to the beginning? That was my, no, all the way back. All right. Which one do Okay. All right. So I'm going to talk about exosomes from the dermatology point of view, which I think is great. And again, I want to thank Ethan and Byung for having me part of this. So I'm a dermatologist. I have been for 35 plus years now doing dermatology. I have lots of academic appointments, and for those that don't know me, right, number 3 and number 4 up here was my pre-COVID world. Every 4 to 6 months I was in China teaching them, but they were really teaching me. And then if you look at number 5, I am the editor of 2 journals in dermatology, which is really a love of mine now. I didn't really think I'd love it as much as I do it. I get between 1,500 and 1,800 articles a year that my team and I go through and try to figure out what to publish. We took a journal, the Journal of Cosmetic Dermatology was quarterly. Now it's monthly. And we have a big team now that helps me do that. So again, all right, we're doing the middle thing, right? Sorry. All right, so this is not the most fun thing to use. All right, so, and again, I'm being sponsored by them, so you need to know that. All right, Byung showed this chart, all right? And if you look here, this only goes out to 2019. And if you think about where we are in 2023, this Google trending thing is off the charts. There is not one person that comes into our clinics now that doesn't ask about exosomes and what do we do with them and what are they and how do we use them and those kind of things. So I apologize here for a sec. All right, so this is my favorite slide that I borrowed from a very close friend, Glennis Ablon in California. Above the water is what we know about exosomes. as a dermatologist or a plastic surgeon. Okay, I'm gonna put plastic surgeon in my world too for a minute. We are just beginning to understand this technology, and it's going to take the community, as Ethan said, so that we can all speak the same language and understand it the same. We have so much more that has to be discovered in our world, and that goes back to the clinical science part of it and the research part of it in the dermatology and plastic surgery and regenerative world of medicine that we live in, and that's all important for what we have to do. There are, however, some really nice review papers. And again, if you were here, if you were at the meeting yesterday, this is a repeat, but I'll try to be a little bit different. A couple times I'll say, take pictures. If you want to take pictures, I never care if people want pictures, take them. But there are some really nice review articles that have recently been published in dermatology that tells us about what, the trend are for skin remodeling, for pigmentation issues, for scarring, for alopecia. Another one in the journal I edit, what the role is in skin diseases and where we may be able to take this field someday, and I think is really important. I'm not going to bore you with the details of exosomes. This is part of a lecture that I give, and I'm going to highlight just parts of it. Right? And because again, you've heard this term extracellular vesicles. If you asked Michael Gold 5 years ago what an extracellular vesicle, I wouldn't know what it was. All right? It wasn't part of what I learned. And then we know their communications and we know they're gaining increasing attention, and again, how we use them and what they are. So anything in red to me is important, okay? They're extracellular vesicles. They contain nucleic acids, DNA, mRNA, and miRNA, as we have heard before. And exosomes are the smallest part of what is an extracellular vesicle, right? And other things, they're microvesicles and apoptotic bodies as well. And as we just heard from Jan, they're part of almost all our body fluids. Blood, urine, saliva, breast milk, and are all constitutively secreted. And they play an essential role in pathological and physiological conditions by manipulating cellular communications, eventually gene expression. So the potential here for us is massive. What we can potentially use these for in our skin problems and skin diseases and regenerative medicine and aesthetics is endless. We got to understand it though, and we've got to pick it out piece by piece by piece to make sense so that we're doing the right thing for our patients. So why do we need them, right? That's a question, because they give us regenerative power, and that's important. Again, we've talked about this, so we'll skip a couple of things. And we know that the exosomes contain, as I said, growth factors, messenger RNA, microRNA, and they can be separately engineered, as you just heard, and we can use these for our skin or for our hair signaling to enhance what we have and to enhance the patient responses when we do these kind of things. And they're really small. And so again, the exosomes here are in the green. And I think that we just saw that in the previous talk, but these are really small targets or things. All right, this is the most important slide I show. I showed it yesterday. I show it in every talk that I do. So if you're from North America, our rules are totally different in how we can use exosomes than probably the rest of the world. So I read most of this slide because I think it's important. The FDA, friend or foe, they are my friend now after 35 years of doing clinical research and having a father, some of you may know, who was a infectious disease doctor who basically did the rubella vaccine, the first influenza vaccine. He developed a drug that you may have heard of called cefazolin. And so the FDA became part of my life, and it's there for safety. Everything is safety. Efficacy is second. But safety is why they're there. So the FDA regulates regenerative medicine products. These continue to be broad marketing of unapproved products considered regenerative medicine therapies that are intended for the treatment or cure of a wide range of diseases or medical indications. And this is exosomes we're talking about. These products require FDA licensure approval to be marketed to consumers. So before approval, they require FDA oversights in a clinical trial. These unapproved products, whether recovered from your own body or another person's body, including all these other parts of things, and exosomes are in there, and they had received before 2019 reports of blindness, tumor formation, infections due to the use of these unapproved products. So every company that was involved in development of exosomes received this letter. And that basically stopped almost everybody in their tracks from using these unapproved products as drugs, right? They're not drugs yet, okay? So again, we've seen a proliferation of companies in the exosome space. At one point, I counted over 80, all right? And in the United States, I think we have 8 or 10 in the skin space. They are human and plant-derived, as we know, and again, we're gonna talk about only one really today. So we heard from Byung at the beginning, there's some important things that are really important here. Cell line development, scalability, upstream cell culture, downstream purification process, really good quality control, being able to have a shelf life that makes sense, all the regulatory conditions, and again, where they are derived from. And again, there are different things that you can derive from, as we all know. We'll focus on the mesenchymal stem cells mainly, and this partnership that brought us all together, right? And so we all believe in what they're doing, mainly because they're doing something correct, at least in the eyes of us, but also in the eyes of the FDA. And again, in North America, it's a different ball game than the rest of the world, but it's also it's where everybody needs to look to for safety and all this other stuff. So again, but this is also an important thing. This is for North America, right? These are post-care topical use cosmetic solutions. In North America, we cannot scalps or any other part of their body. It's against the rules. So we are stuck using these topically. Again, that's not a bad thing until we have the science and until we have everything, until they spend the money to create a drug, we can't do this. The FDA can come in our offices if they find out and shut us down. They're not going to shut the company down, but they can shut us down because we're doing it. So they actually go by the International Cosmetic Ingredient Nomenclature Committee, and the name human adipose stromal cell exosomes was given and published by the International Cosmetic Ingredient Dictionary, which is the big book. And these products are not drug products. They are not intended to prevent, treat, or cure diseases or medical conditions, and they are not intended to be injected or delivered intravenously. So if you're a dermatologist, like I am, they are not intended to prevent, treat, or cure diseases, right? I cure a lot of diseases, but I also treat a lot of diseases where we have no cure. So dermatologists are really good at treating things. I can look at a rash, I can look at a condition, I know how to make it go away. It may not cure it. So I'm pretty used to that. And I think we all have to get that behind us and understand that. But again, they are not to be injected or delivered intravenously. I will repeat it 100,000 times. All right, we were there this morning, the US contingent and the Canadian contingent. There's now a second building going up because they have run out of space. Again, 55 patents, 8 US patents, 16 published papers. I run it to journals. I want papers. I want to see the science behind this. Unfortunately, in the US, we want the US data, right? And again, I look to Ethan and I go, thank you, because we're doing it, right? It has to be done, and it has to be done a certain way. And we know from all the work that they have done before, this stuff should work in a lot of things that we're interested in, regenerative and aesthetic medicine. We know it increases elastin. We know it increases collagen. We know it increases human fibroblast cell proliferation. That's the bench work that we've been able to do, or they have been able to do, to get us where we are. And we can combine it with devices, and we're working on proving that this combination is really special. And again, I think you're going to see a lot of cases today of lots of different combinations where this works. This is the only paper in aesthetic medicine that we have, right? Period. There is none. Again, we have to do this in the United States. And again, we were talking before Jan and I were talking, this is the split face, double blind, placebo, randomized control trial. Hard to do in the US to get ethics committee approval to do these kind of trials on the face. That can be done off the face pretty easily. But again, this showed better efficacy and shortened treatment time for these patients. And so when you look at patients, and I'm just gonna go quick in the last couple minutes, all of these patients have been treated with a procedure immediately post topical application of exosomes, the ExoCoBio and Benev exosomes together to give us a better clinical outcome, right? And time again, we shorten the downtime. We think we are enhancing the cosmetic outcomes. We know we are, but again, we're going to prove that. And everything gets better faster. And so, you take a patient like this, and I can put this patient out for 2 weeks with what I can do with some of my devices. And if I can do this and put them only have downtime for 1 or 2 days, they're gonna love me, right? And they're gonna get great results when we do this. Again, using some, pretty aggressive techniques, but again, we improve everything, make them better, faster, and that's the goal of what we're doing. And again, the clinical work shows that we can use this both on the aesthetic side, but this is more of a medical condition, right? Post-procedure, radiation therapy, burning and irritation, we can do this. And then again, wounding, and this affects all aspects of the wound healing cascade, and we can heal wounds really fast. And so that's where it gets important, again, with more of our procedures, with exosomes on one side, without exosomes on the other. You see the decrease in erythema. That's what we're looking for. The improvement here in the acne scarring post-procedure. Again, this was a skin necrosis. Again, I would never have thought of using exosomes in this kind of situation before. We're good at healing wounds, but now I think I'm better at healing wounds, and that's important to me as well. So again, really bad acne scarring, doing our procedures, healing the skin a lot faster. And again, we think that the exosomes are helping in that cascade of wound healing post-procedure with our needling or with our CO2 lasers to make our patients better. So everything gets better with this. And I'll end with one of my patients who thinks this is the best thing that they've ever done. And that's what I wanted to share with you today. So thank you all very much. And so now, and we're going to have a question and answer after the second session. So if you look in your program, there's 2 sessions, and then we'll have a question and answer, and then I think we'll have dinner. But now it's my pleasure to introduce a very good friend. Dr. Renato Saltz is a plastic surgeon based in Park City, Utah. He runs a bunch of different meetings in the United States, and one is the Brazilian meeting. It's a Brazilian-American plastic surgical group, but it's in the United States, right? And then he also is involved in global aesthetics, right? Which is in Miami. He's been part of other things that I've known for years. I've watched his incredible work and teachings for a long time. And he's gonna talk on aesthetic surgery and cosmetic medicine strategies for success. He's going to tell us how to do this in a practical sense. So Renato, you're up.
Thank you very much, Michael. Really hard act to follow. I didn't plan this, but Ethan mentioned The Tipping Point, which is one of my favorite books. And my wife and I read it many times. And what he did not mention is a chapter that talks about Paul Revere and his importance during the English invasion or attempt to take over the United States early on. And in that chapter, Malcolm Gladwell talks about connectors and leaders and why one person can do a lot of things that many others couldn't. And that's how I see you, Ethan. You're a very unusual CEO. I know a lot of CEOs. I know Michael knows a lot of CEOs. We work with them. And you just came to the US scene and start bringing people together. And always in this very humble, friendly way. And so I congratulate you the way you do things. And now that I met your partner, Mr. Cho, I can see you guys work well together. So I really appreciate what you've done and introduce us to this world of exosomes and Silvermax. And what I'm gonna do is show you or share with you how these things work in my practice. And I'm sure there are a lot more smart business people in this audience, and I'm sure you have many different ways to do this, but it's working well in our practice in how we incorporate all these things, surgical and non-surgicals, in a busy aesthetic practice. So, I'm sorry. That should be the end of it. Okay, I got it now. I'll go with the, so this is actually this is part of a whole book about mixing aesthetic surgery and cosmetic medicine, but I'll try to focus and do this in a short period. I'm double boarded. I'm board in general surgery and plastic surgery, and I've been involved with societies pretty much all my academic life. And it's a good thing because it exposes you to a lot of different people, different countries, different modalities, and you learn to appreciate what is done all over the world instead of just focusing in the United States. So another reason I'm here, I wanted to learn more about what an exosome is, and just listening to my colleagues yesterday, today, and hopefully tomorrow, it's been a wonderful trip. So thank you for bringing us. This is my disclosure. I'm involved in several advisory boards, and I think it's great. I really push my residents and my fellows and colleagues to get involved with the industry. In the 80s, when I was a resident, was a bad thing. And I think it's very important because working with them, we get better products and better treatments for our patients. So that's what I do, and I published 7 books in plastic surgery. Okay. If I can get this going, Mr. Chairman. Little button on the mouse. Okay, here we go. So we're talking about exosomes, we're talking about disruption. And disruption has been great for plastic surgery. Disruption has been a very important part of what we do, and it's not necessarily a bad thing. Disruption has happened to our patients in aesthetic surgery, especially right after my training in the mid-90s when neurotoxins became popular. And the patients are very aware of all this. The patients want to have procedures done. This is really not going. Okay. Patients are getting old, and not all of them want to have surgery. So they want to remain looking young. Okay, go this way. The timer is still going. And I know he's very tough with the clock, so. Okay, here we go. So patients are getting old, and they will do whatever they want to do to remain looking young. And surgery sometimes is not the first option. When you look at the statistics, especially from the aesthetics side, which is the best statistics in surgery for us, you notice the red line is surgical procedures, and the green line is non-surgical procedures. So the surgical procedures are growing at a very different pace in the United States. And then you look internationally, this is the last one we had from the ICEPS, our global survey. Look at the number of procedures, they continue to decrease every year, while the non-surgical procedures continue to go up every year by 10% or more. So, I'm sorry. Here we go. So in 2002, after spending 13 years as a full academic professor in plastic surgery, I realized I gotta do something about it because the University of Utah wouldn't let me open a medical spa, wouldn't let me buy devices or anything different than traditional surgery. So we went into private practice, and the goal was to deliver pre- and post-surgical care with medical spa treatments under the direct supervision of a board-certified plastic surgeon. So, and why I did that? Because I always follow this statement that if you don't change, if you don't modify your what you do, most likely you're not going to survive. And that's true also for our specialty in aesthetic surgery. So we opened two practices, one in Salt Lake City and one in Park City. And that small hospital is a very nice boutique hospital in Park City. It's a beautiful place in the mountains. Come and visit us, and you're going to see a totally different way to deliver excellent medical care in the mountains of Utah. In this, you know, we can spend hours talking about this whole presentation. I'm going to focus just in some areas where the non-surgicals play a very important role, and I'll include Silvermax, I'll include exosomes in that part. This has been published in 2012 in this book that we published with some excellent co-authors that covers all aspects of this talk. So the facial aesthetic package was created in the office for to bring together our patients, our practice. This is really not working. Let's see this way. Yeah, the other way. Here we go. I'll learn. The facial aesthetic package was just a way to create a system that staff, patients, everybody involved in the patient care would understand how we do things. And I'm sure you have different ways to do it. And it's divided in three components: the pre-surgical component, the intra-surgical component, and the post-surgical component. So all these patients that come in for, let's say, a facelift, they're gonna go through this. And everything starts with the consultation. And to summarize the facial aesthetic package, you can think of, she's coming in for a smile and she gets the whole treatment done. And that's how this concept is better explained to patients, because I know if I don't offer, she's gonna fly to Nashville and Michael is gonna take care of her. So you wanna keep the patients with you, better be ready. And this is a perfect example. This is a very beautiful lady. She was 63 or 64 when I first saw her. We did a whole preoperative preparation. Then she went through surgery. And then postoperative, she continued to be seen by our master estheticians. And you can see here, 4 years later, she's still looking very good. And I see her maybe once a year for pictures, but she comes every 6 months to be treated by our master estheticians in all these non-surgical procedures. And this keeps these patients in the house, keeps them very happy, and it's what I call patients for life. And we'll talk a little bit about this later. So it always starts with a consultation. And that's really my important part of the whole process. And the consultation is really a team approach to educate and treat patients in all aspects of aesthetic surgery and cosmetic medicine. And one of the things I really like, and you are welcome to take a picture of this, is a treatment plan. And the treatment plan is outlined by the surgeon. So I have this consultation with a master esthetician next to me, my PA, or my nurse, and that's when I outline what this patient's gonna do. So look at where Silfirm and many times exosomes is part of the treatment for scars, for skin laxity, for wrinkles, for hair loss, for crepey skin, wrinkles, fine lines. You know, it has become an integral part of what we do. Post-operative, these patients come back and they have all these treatments, as I was gonna show you later. Another important aspect of a successful marriage, successful combination of surgery and non-surgical procedures is, what do you have? What do you offer in your practice? So I'm a firm believer that you have to be innovating, you have to be competing, you have to stay ahead. And if there's a new exosome in Korea, I want to know. I want to learn about it, and I'm learning a lot. And that's what plastic surgeons should do. And we carry a lot of stuff. We have 2 medical spas. They're very busy, and we have patients coming from all different states. So you need to be ahead of the competition. So when Silfirm became available, and we had Ethan and Dr. Na coming to visit us, and was a very interesting couple of days in the office, because we have the demonstrations by Dr. Na, and then after the treatment, he showed this, let me see if I can make this video work. He showed this little vial, and you have volume? And this magic little vial, and
decrease healing time.
so Megan just had a treatment with the Silfirm by Silfirm's creator, Dr. Na from Korea. And now Patty is applying exosomes. Here's Dr. Na. Thank you for joining us. Thank you for coming. Welcome to Utah.
My pleasure. Yeah.
Megan loves it. Look at her face. Say something.
Well, I really love the massage, the after massage from Dr. Na. That was the best.
My goodness. Okay. So now you have to stay with us. You're a new member of the SALS team.
Thank you so much. You enjoy new technology of first wave with Silfirm Max and Dr. Na's special massage together. Thank you.
So this was it was totally news to me, and from that first visit, I started learning more and more about this product, and which brought us here today. So in the last 2.5 years, we've been using several combinations of Silfirm Max with exosomes. These are some of our own patients with improvement, and you're going to see a lot of case studies today. So I'm not going to bore you with this, but it's been quite incredible. What I like the most is when we combine in our surgical patients. These are patients that come in for facial rejuvenation, and we get them right on some of these treatments. And you're gonna see some incredible results here, which is not necessarily because of the surgeon, but because of all these non-surgical. This is a 74-year-old patient for full facial rejuvenation. Underwent at 3 weeks. She sent me this picture. She was going to vote actually in November. We're all very surprised with the amazing preliminary results she had. But she continues to come back. And here she is at 6 weeks, and we started immediately back on the non-surgical treatments, especially Silfirm Max and exosomes. Again, this is all subjective. This is not scientific. And here you see her at 1 year. She continues to look better and better. I mean, this is amazing. This is not your typical facelift result, even by salts at 1 year. So the credit goes to all these non-surgicals that she's getting. And if I could just advance this. Here we go. This is another patient from Newport Beach. The post picture she sent to us. She went to see a colleague there for the post-operative picture at 6 weeks. These are great results. Continue with that, radiofrequency microneedling and exosomes. And these are just some of the patients we're now using and getting this kind of results with improvement in the quality of the skin in their 40s. In their 50s, used to be a smoker, New York, a lot of sun damage, and we're getting this improved results that it didn't happen before. This is a 60-year-old from southern Utah, a lot of sun damage. These are difficult cases because you have a skin that doesn't respond well to standard treatments. So we're treating them now with a lot of radiofrequency, a lot of microneedling in aggressive exosome topical treatments, as Michael pointed out. And I have a couple more if I can get to advance this. Maybe we need new batteries, gentlemen. This is a 7-year-old from Arizona. Again, a lot of sun damage. And we're just getting better surgical treatment than used. And age is not a factor as long as they're healthy and the anesthesiologist is willing to put them to sleep. I mean, she had a full facelift, brow lift, and again, aggressive treatments with non-surgicals in the med spa. And the concept of patients for life, and I'm finishing, Mike, it's a very important concept. I want you to take this home. These are patients that will stay with you and your team until they're ready for surgery or continue non-medical procedures. And I think that's where non-surgicals play a huge role. This is a facial rejuvenation surgery combined with med spa treatments, age 52, day 4, at 3 years. These are all full facial rejuvenation. Patient from Hawaii with a lot of sun damage at 1 year, and now she's at 3 years out. These are happy patients. They fly in just for treatments. There's a patient from Baltimore, East Coast, 66, 1 year post-op, and now she's at 6 years. Notice she looks better every time. And you know, that's not just the facelift. That's a lot of the non-surgical treatments that make the difference. And here you see her neck. This is a 60-year-old, 1 year, 8 years later, she looks better every time I see her. The quality of her skin looks better. You know, it's just a big change for our surgical practices. And I have a couple more. This is a 12-year follow-up in this patient. I revised her neck at 10 years, and it looks like she's not aging. And look at the quality of her skin. It's just amazing. And this is a real long follow-up, 13 years, patient from the Middle East. She flies once a year to Utah just to get medical non-surgical treatments in the office. I see her, I take pictures, and you know, very happy. So in summary, the bottom line, which has done besides this very happy patients, has also brought a lot of money for us. These are when we first brought the 2 machines to the practice. This is the first year in 2020, 3 months. Then this is the second year, 2021, and we started using exosomes at that time. 2022, you see the numbers keep going up. Now we're doing, in 2022, we started using more and more for scar treatments. And this is 2023, the first 6 months. You see the numbers there, so probably double before the end of the year. So that's the bottom line. That's what generates in our practice, and these are true numbers. So I want to thank Ethan, Sunil, and Diane for introducing me to the exosomes. It's been a big impact in my practice, and this is terrible. And I want to thank this group for, if I could go back, this group for their knowledge and research and advances in our field. And finally, thank Ethan and Mr. Cho for inviting us to Korea and show us their facilities. Thank you very much.
Can I get this on? Thanks, Renato. That was great. So we're going to move on. Again, we'll have question and answers in a little while. So one of the nice things about being old is I actually get to sometimes introduce somebody who spent a lot of time at my office while he was in training and watch them grow into superstars. So our next speaker is Inigo de Felipe, who is from Barcelona, Spain, who is going to talk about exosomes in cases of inflammatory scalp conditions. So Inigo, you are up.
I understand why this was not working. So guys, when you come up, backwards is forward and forward is backward, okay? So it should be like this. I was a medical resident when I met Michael for the first time. That was in 1996, because I asked the manufacturers of a laser company, right, who's the best guy in IPL and lasers in the United States? And they said, clearly, it's Dr. Gold, you should go there. So that's why I've met him, and I've always tried to emulate him and to do things as him. Obviously, he's much more successful than me, but I think he's a model on how to be. So thank you very much for being here. Exosomes in the hair are actually very, very effective. And you've seen my presentations in the previous years. And let me focus on this guy here. It's the one in the center. I don't know how to point this. Okay. So you see this guy here, let me just come here. This one, right, was treated at the end of July in Ecuador. And this is the picture they sent me 2 days ago. So you clearly see that with just one session of exosomes that were actually injected, not like what you should do in the United States, but this was Ecuador. They're more relaxed about rules there. You see the great effect of just one session of exosomes. So what happens when we do this in inflammatory conditions like alopecia areata and so on? Okay, yeah. So, and let's talk about what happens in all these conditions and some things that we've learned with this. All right. It takes its time, all right. Yeah, all right. So, alopecia areata is an inflammatory condition of the scalp that thankfully is not scarring, so we're not destroying the hair. In this situation, many follicles go from growing in anagen into telogen. It is diagnosed clinically. We don't really need a biopsy for this. And it's very commonly associated with other inflammatory conditions, not only of the skin, but also outside the skin. It is associated with vitiligo. It is associated with diabetes, with hypothyroidism, and the treatment usually is corticosteroids, applied topically, but probably the first line treatment is to inject corticosteroids. Some of these cases are very resistant, nevertheless, to this treatment, and JAK inhibitors have been introduced, but they're not all that easy to get. At least in Spain, it's very difficult to get JAK inhibitors, only in public hospitals. So patients sometimes are desperate to find solutions. So in alopecia areata, there's clearly a problem with lymphocytes. And these lymphocytes of the CD8 type, they infiltrate the follicle, and they start doing something to the hairs that we don't fully understand, and these hairs react by sleeping. Sorry. So what these guys did a long time ago was, okay, let's see who's responsible of the problem. Is it the T lymphocyte? Is it the follicle? So usually in alopecia areata, what happens is that not in alopecia, sorry, in normal hairs, these hairs never show HLA receptors. So they're really not immune stimulating anything. But in alopecia areata, funnily enough, these hairs show different receptors that are found by our immune system that starts attacking this hair. So what they did was, okay, let's transfer hairs from a patient with alopecia areata to mice that did not have an immune system. And what happened was that there was no alopecia areata at all. So what they did again was, okay, let's transfer as well T cells, the T cells of the CD8 type, and still the alopecia areata did not happen. So they needed to transfer everything. 1, the hair; 2, the CD4 lymphocytes; and 3, the CD8 lymphocytes in order to reproduce. So what we know today is that everything is needed in order to produce this reaction. So clinically, and this is very important, right? There is a lot of ICAM1 receptor in the lymphocytes that enhance this reaction. and makes us develop alopecia areata. And you can see that with this stain, up there you see patients with alopecia areata, down you see patients being cured, right? They express all these receptors that are found by the lymphocytes. That's why they attack the hair, and all these hairs go into telogen stage. If you follow clinical recommendations, right, and this is what they recommend general practitioners, this is not for dermatologists, but you'll see how funny this is, right? What a GP or a general practitioner should do with a patient with alopecia areata. So the first thing would be, okay, if you don't have a lot of alopecia areata, there's less than 50% of the first surface affected, the treatment options are there. Topical steroids or minoxidil, or the first line treatment, which is injections of steroids, usually injections of triamcinolone or Kenalog. Okay, so that's option number one. And I think it's very important when we talk about new treatments like exosomes to focus that there is an order. I mean, if you have a wife, you have to invite her for vacation, not another girlfriend. Okay, so there's an order in life. You understand this, right? So if there is a treatment that has been proven to be effective, you have to follow that. So exosomes should be a second line or a third line treatment if they are approved for this treatment one day. I think Michael will help us in guiding us for that. So the funny thing here is that GPs are told this, okay, if these people don't respond, right? Or if there is more than 50% of hair loss, the treatment options should be, okay, consider oral prednisone, or, and this is actually very interesting, refrain these patients to the dermatologist, right? Refer them to the dermatologist. They will know what to do. Right. So this is the list that we dermatologists would have to do in a second line treatment. Okay, we can still apply topical or infiltrated corticosteroids, or we can go to the difensoprone, which is a very old treatment. We can do LED, we can apply JAK inhibitors or take JAK inhibitors. So exosomes are not in the list, but because we like trying things, all right, and we know that exosomes derived from adipocyte stem cells are very effective in pathologies related to this, like eczemas or atopic eczemas. What we did some time ago was, okay, let's treat patients that did not respond to these first line or second line treatments and see what happened. And this is one of my first cases some years ago, right? This woman has been suffering from alopecia universalis. You say it's universalis or universal when it affects not only the scalp, but also the eyebrows and the rest of the body. And she didn't respond to topical, to intralesional, to oral prednisone. So we had this option, which was exosomes. We talked with her. This is after 6 months. And that last picture there on your right is after 1 year. So after more than 1 year of that picture, we don't have any relapse. This is another guy. He's had 6 sessions of exosomes, and when he came here, right, he did not respond to oral cortisone, he did not respond to topical, he did not respond to intralesional, and we did exosomes. And this is another person. And as you can see, this is not a universal alopecia areata, but because some patients want to have something stronger and faster, we offered him exosomes. And as you can see, he has not only improved from his alopecia areata, but he's also improved from his androgenetic alopecia. And you can see here a very interesting recovery of the hair in the frontline area. And we have many patients that have been treated like this. The only question is, well, there's not only one question, obviously there's several, is whether this may have happened without any treatment. And that's the first thing that might occur, right? And the second one is what would happen if we injected corticosteroid. So we are now going through a study in which some patients are assigned to a treatment with infiltrations with cortisone, with Kenalog, and another group is being treated with exosomes. We are comparing, and maybe in one future meeting, if I'm invited, which I suppose I will, I will share the results. So as you can see, right, there's not only a recovery of hair, there are other changes. And I would like all these clinicians in this room to tell me, what do you see that is happening when you treat patients with alopecia areata with exosomes injected? So what's happening? Yeah, exactly. So there's darker. So my first thought is, okay, it's darker because the hairs are thicker. But it's not really all that much. I mean, you can, not in this patient, but you will see afterwards in some patients, there is clearly a reduction in the number or the percentage or the proportion of white hairs in the total hair. And the second thing is that it's very effective in the frontal area. And when we do treatments for androgenetic alopecia, usually they're very effective to increase the density of hairy areas. But when we have these areas here that are naked, we're not really very successful. But in this case, exosomes are really very effective, and you'll see some patients afterwards. So because this is effective in alopecia areata, if we change subject and we move into lichen planopilaris, which is not all that common, and it's very difficult because the hairs have been destroyed completely, right? We have tried. This is after 1 month of 3 sessions of triamcinolone, and you can see that the patient is not responding. On the left side, you have a patient with folliculitis decalvans, and I treated him with 1 session of triamcinolone, 3 sessions of triamcinolone plus exosomes, and you see that the result or the response was much better. So maybe there is a potential effect of exosomes that we cannot get with Kenalog. And this is actually the trichoscopy of the previous patient. You see on the left, you see the areas, the white areas with the disappearance of openings. It means that the hair follicles have been destroyed. And on the right side, you see that the patient did not respond with triamcinolone. So when I ask myself, because we all have to make questions to ourselves, right? If patients receive exosomes and they improve, would they have improved if they didn't get exosomes? Well, this is the response. They didn't. I only have few cases for this, but it seems that exosomes really potentiate and improve the result that we're getting with other things. And in fact, this is one picture that Bjorn showed to those people visiting the factory yesterday, and you see an improvement in a scar that would never improve with anything else, and I've been practicing medicine for 29 years as a dermatologist. But he forgot to show the final result, which is this. You see where the arrow is, right? So there is a complete recovery of normal skin where there was a scar. And this is after 8 infiltrations of adipocyte stem-derived exosomes. And this is actually not happening with any other procedures. And I've been working with CO2 laser and with erbium laser. I promise you, you never get these results. Only exosomes can deliver this. So because this is working in fibrous tissue or scarring tissue, we can now move to a 4th indication, which is frontal fibrosing alopecia. In this case, we not only have the inflammation, we also have the scarring. We have the disappearance of the follicles. So it'll be very difficult to really create new follicles, right? So what happens when we have a patient like this? This is the typical trichoscopic finding. That's the typical biopsy with a band-like infiltrate of lymphocytes. So what happens is that it also works apparently, but this is only an evaluation from a clinical point of view. It's very difficult to count exactly how many hairs she had at the beginning. But obviously, this lady, this is after 1 year, improved clinically, and this other improved. And what's more interesting, there is no recurrence after 2 years. Sadly, we don't have enough experience with these cases because I'm running. We're running out of time, but let's do it a little bit in the Spanish Latin way as well. So there are two things that are not related to inflammation, but they're related to something that we're learning, which is the behavior of the frontal temporal area, which is very difficult to respond in other treatments, and the response of graying and whitening of the hairs. So let me just focus a little bit on this. This is a study in which they compared dutasteride against finasteride. And as you can see, the important thing of this study is not really that dutasteride is more effective than finasteride. We all know this, right? What you can see is that the response in red, that's the frontal area, is much poorer than the response in the vertex other areas. So constantly, and this is what we're seeing with all the treatments, the frontal area does not respond as well as the rest. So when patients come to me, they say, "Doctor, can I recover the hair here?" Maybe the only answer is you have to go under hair transplantation. But maybe with exosomes, this changes. And it changes because we're constantly seeing a recovery of the hair in difficult patients. That's the trichoscopy. And you can see these areas of trichoscopy, they were taken more or less in the same place. We measured with centimeters, and there is a recovery of the density of hair. So that's the first thing that we've discovered with exosomes, that they are very effective in this area, whereas other treatments are not. And the second thing is the graying, and I'm finishing with this, but I think it's probably the most interesting topic of my lecture today. So we know that with time, hairs lose their pigmentation. And it was said that it might be related to mitochondrial DNA. There are many papers about this, but actually the content of DNA in mitochondria does not change in white hairs in comparison to normal hairs. At least this is what experts say. So when we treat patients like this, we really discover that there is an increase in the color of the hair. So we don't know whether it is because we are stimulating hairs with color or because the total proportion of dark hairs is increased. We don't really know what it is, but obviously all patients say, "Hey, doctor, my color is coming back." So this is one case. This is another case. This is a very interesting case because this man, and this is on the left, had an exosome plus nanofab plus LED plus dutasteride, finasteride, and minoxidil treatment, and this is how he appeared 8 months later with just one session. So he came back from Switzerland, and this is where he came, and he wanted to do another session, and I just injected PRP, and he didn't get much of an improvement afterwards, right? But the reality is that the color of the hair improved. And this is another case. And this is another case with just one session. She only had one session of exosomes, and you can clearly see that the proportion of white hairs is lower, or the proportion of dark hairs is higher. So this is not the first time that in the history of medicine we discover that white hairs are reduced or they recover the hair. It has happened with different treatments. It's very anecdotal, but you can see with ribavirin, interferon, one patient with pazopanib, I don't know what type of medication that is, with levodopa in a patient with Parkinson, with thalidomide, and with these medications, lenalidomide, I can't even pronounce them, they're really difficult. But this happens. This is, for example, a case treated with thalidomide, right? This is one case after several years. And this is a series of 14 patients published by some friends of mine in Barcelona, right? And this is what happened to some patients that followed a treatment against lung cancer. And what was the treatment about? It was with some agents, nivolumab. I don't even know that these medications, these drugs existed. But these drugs are interfering with a pathway, which is the programmed cell death pathway, which I will show now. So what really happens is that this is what would happen in the situation of a cancer cell. And this is probably the most interesting part of my lecture, so just listen to what I'm saying. Not all exosomes are exactly the same. This is a cancer cell, a tumor cell, and that's a lymphocyte. So supposedly lymphocytes have to defend, have to protect us against these tumor cells. So they have to kill these cells. And what happens is that once the tumor cells receives the interferon, right, this tumor cell is going to produce some exosomes containing a dangerous thing called PDL1. It's called cell program ligand that will activate the death of the lymphocyte. So therefore, if the tumor is attacked by our lymphocytes will die. It's quite dangerous. So not all exosomes are good. So if we stop this pathway, we will enhance our immune system to attack and destroy our tumors. And those medications that increase the pigmentation of hairs actually were interfering this pathway. So basically this is the drawing that I was making yesterday for you, right? In normal immunity, we have this programmed cell death ligand. So what happens is that a lymphocyte will produce interferon gamma, which, by the way, exosomes from adipocytes will reduce. And the melanocyte, for example, right, will develop this glue called ICAM, right? And with this ICAM, they will kiss the lymphocyte, and the lymphocyte will die. So our system, right, our melanocytes are protected from being attacked thanks to having this programmed cell death ligand, right? If there is a mutation or something in here, right, which happens in autoimmunity or in vitiligo, for example, melanocytes with vitiligo do not have this PDL1, right? So what happens is this. The lymphocyte produces interferon, we produce ICAM, and who dies? The melanocyte dies. That's why we have vitiligo. Now you understand? So what would happen if we use exosomes? When we use exosomes, we reduce the amount of ICAM1. So we stop the kissing between these two cells. So this one lives happily together with the lymphocytes because they're not attacked by each other. And I think this is probably the way why exosomes increase the pigmentation of the hair. So just to finish, I'm going to throw to you two questions. I'm not going to tell you the answer, but these are the questions. So this is a picture from yesterday. Out of these two guys, right, we know that this big guy is younger than me, and that lady, Julius, is very young as well. But out of these guys, who is older and who has had more exosomes injected? So thank you very much.
All right, thank you, Inigo, very much. So again, part of this is to stimulate conversation, and I think with Inigo, we get some great conversation and thoughts, right? Again, outside of Spain, and if you're in the US, you can't do this. All right, so next up, Dr. Rosario Salud Abaya Blas is going to talk from the Philippines, is going to talk on exosomes in medical dermatology.
Hello, thanks for inviting me. Thank you, Dr. Gold, for that, and thank you to ExoCoBio for this invitation. Sorry. Next slide, please.
There is you can use the roller.
Thank
or you. Can try the clicker. And at the break, we're going to fix this.
Thank you. Okay. I just have to wait. Thank you.
Silly.
So every time I speak about exosomes to anyone, I always tell them that the learning curve is not really in what procedure you're going to be using to introduce the exosomes. The learning curve is in finding the answers to these basic questions. In particular, where did the exosomes come from? How it was made? Are there studies using that particular product? And for me, if the product is registered in the country of origin. Now, in my practice, I normally apply the exosomes topically. Dr. Gold will be happy to hear that. And I use microneedling deliberately because I want to see the exosomes do all the heavy lifting. And I try to refrain from energy-based devices. Next slide, please. There. So now, you can take pictures of anything except for the patients. This is a patient, an 88-year-old patient who had bed sores, and she's been around the block. Plastic surgery threw her back at me because they had done everything they could. And the only thing that I had in my armament was just the exosomes because I had just arrived from Seoul last year. In 7 days, just applying the reconstituted vials, in 7 days, the patient had healed. She had very good nursing care. So nursing care is indispensable. So I had the patient's caregivers apply the reconstituted exosomes 3 times a day because there was a paper in China saying that 3 times a day was the requirement. But knowing us Filipinos, Asians, they really don't follow it. So twice a day was the minimum, but it really worked. Now that's my other patient. My other patient spent a miserable Christmas because he was drunk and then he had something, a metal rod impaled in his leg. He went to the ER, they sutured it, but it dehisced. So he went around limping until New Year and then he was referred to me. And I said, oh, I can heal that in 1 week. From my experience with bed sores. It took a little bit longer than 1 week because my patient was apparently a vapor, a cigarette smoker, and he just didn't want to admit and stop his habits. But exosomes did the hard lifting, did the work, and in 12 days he was fine. Next slide, please. And this is another patient who had ameloblastoma, and the wound, the donor site, never quite healed. So he was walking around with a painful wound that never quite healed for about 2 years. And so he was referred to me, but since the patient was in a hurry to get home, I just reconstituted the vials for him and applied it and taught him how to do it. And he went home with it, and in 7 days, you can see that even the skin around appears better. And that is one of the reasons why, that slide over there, a new article, exosomes will affect positively all the stages of wound healing. Next slide, please. And this is one of our favorite patients. This is a 69-year-old patient who came in with exfoliative dermatitis. New Year was very bad for him. He had a history of atopic dermatitis, and his well-meaning daughter gave him some herbal medicine to apply, and he burned. So basically, he went around the block, oral steroids, topical steroids, changes in skincare regimen, everything, before he was finally referred to us. Within 20 minutes, after the application of reconstituted exosomes, the patient said he no longer felt any burning sensation on his face, and the redness had begun to disappear. And you can see it even at his back. Next slide, please. And he spent a happy Valentine with us. And this photo was taken 4 months after the last application of exosomes. Of course, we had to tell him, don't apply anything else. If you want to apply anything else, let us know first, or else the problem would come back. Next slide, please. And these are my other cases. Lichen simplex chronicus, a patient with COPD, diabetes, and he was overweight, and he's been already through so many things, steroids, changes in moisturizers, microneedling at the depth of 1.5 millimeters. And just 5 times I applied it, 1 week he's better. And that's also hand eczema, a depth of 0.5 millimeters, 5 times, apply the exosomes, and we're done in 7 days. Next slide, please. And this is a patient that had endometriosis and a wedding to attend. So after 3 sessions, she was ready to say, I do. Next slide, please. And this is another patient. Again, my patients have a tendency to give me deadlines. So the deadline for this patient was 1 week. I made it up to 10 days. So just 1 session with microneedling 5 times with the exosomes, whatever is left in the vial, I sent it home to the patient in an insulated bag, just the way Dr. Duncan did. Excuse me, Dr. Duncan did. Because in my country, we don't have ExoBalm readily available. So I just made use of whatever the patient had. And the patient healed. Next slide, please. This is a patient with alopecia areata, but you've seen better pictures with Dr. Inigo. And that patient on your right side, that patient has undergone a series of autologous micrografting. And usually when I do autologous micrografting, I normally see the patients begin to lose their hair at 1.5, 1.7 months. And so this patient wanted a better option. So what you see on the lower side, that's after 4 sessions, that's on the 3rd month. I normally will see that usually in a patient that has had autologous micrografting around the 6th month. And the patient's very happy. Next slide, please. And that is it.
Thank you. Thank you. Those were some great cases. Again, I think what you just saw in the last 2, the inflammatory role of exosomes is going to be very important. And maybe we'll develop a drug at some point to be able to actually use these injectable in our part of the world. We're going to have one more talk in this session. It's not in your program. Dr. Gabriel Buendia from Spain is going to come up and talk on his experience as well.
Okay. Thank you so much for having me at the last minute. I just wanted to share with you a special case report of treatment we did and some experiment we did with a patient. So what happened is we did a really unforgiving CO2 fractional laser on a 53 years old woman that had never had laser procedures done before. Very high energy with Fraxis CO2 from Melota. And then we went just crazy and applied exosomes only on one half of the face. We in Europe have the vegetal version from Rosa Damascena that is giving us amazing results. And let the pictures talk for themselves. That's 24 hours later. You can really see a clear difference between the side where you had exosomes and the that didn't have exosomes, both in the bleeding, in the erythema, in how the wound healing is happening, the definition of the microthermal zones and the healthy tissue. Here's some details all over the face. But we can go further. We can see the volume difference because of the edema. The edema is completely different on the exosome-treated side compared to the non-treated side. So that's only 24 hours. And, the erythema, some more details. Even we can use the pigment analysis to see more clearly the difference between the healthy skin and the scabbing on both the exosome sides and the non-exosome sides. The non-exosome sides look like a mesh. And if we go with the UV fluorescence, we can even see more clearly. You can really see the wounds and the healthy tissue in the deeper layers on the UV side, and you can see a mesh of lesion on the other side. So by the way, a very quick reference for UV fluorescence. Very clear blue is healthy collagen. The bluer it gets, the healthier and younger it is. Grayish blue means elastosis, so it means sun damage. And this kind of yellowish, greenish fluorescence normally is what happens with healing. You have the elastin that has the fluorescence that normally is hidden by the collagen that you can see, pro-collagen. So you'll thank me for this later. So this was day 2, 48 hours after the CO2. Similar to day 1. Apparently, but when we compare on the edema side, it's even more striking. Only 2 days after the making of a savage CO2 fractional laser, almost all the edema has disappeared on the exosomes to the side. And then we changed plans. We went to phase 2, and we applied late exosomes in the other half. We wanted to see what late application of exosomes could do in this lady. Could we still help her or not? So this was a Friday, and we had the weekend happened, and on Monday she came back, and there was a striking difference on the non-treated side now. And we compared the edema, and both sides were equal now. So that's very big news. Even if you, for some reason, are late in the delivery of exosomes, you can still help patients with a very strong anti-inflammatory effect. So this is, I think, the first time you've seen that. And well, the wound healing improved on both sides, but the early delivery was still beating it in terms of falling scabs, improved skin texture. And here you're going to see something very, very new, I think, at least on the image side, it's in the UV. You see, you see the yellow, the reference. This is the first image I've seen of early onset of neocollagenesis and regeneration caused by the exosomes. The late exosomes delivery site is healing normally, and the immediate delivery is already having elastin and procollagen happening. This is 4 days after the treatment. We let other 2 days happen. So here are some details. Further improvement, almost no more inflammation. This is 6 days after the laser. And on the UV side, we have the normal regeneration happening on the late delivery, and way more regeneration happening on the exosome side. So everything has happened. So the detail, and this is 21 days after the laser. Full recovery of the patient, apparently similar. And obviously there's a big improvement on skin quality and texture, rhytides, fine lines. You can have some details here on improvement. This is the early exosome side treated. Rhytides, texture, pores, everything, the texture is widely improved. And on the collagen side, you can see it's bluer now. It's way bluer on the cheek where we had the double pass, and still mildly better on the front. It's not an amazing result, but we still have a collagen improvement. And it also happened on the late delivery side. So we thought, let's compare them. Are the results equivalent? And yes, no? Now, let's compare them. Texture-wise, I think if this wants to move, no? Okay, texture-wise, I think the texture is way more improved in the early delivery side. On the rhytides and the pores, both results are good, but the side with the early delivery was still better. Also, in the texture, we beat it. And finally, on the collagen side, we can really see that the collagen regeneration is way better on the side where we gave immediate delivery of the exosomes. We have a very homogeneous blue fluorescence, while on the other side you still have some transitory hyperpigmentation, and well, and you can really see the difference here. So giving an immediate delivery of exosomes does the heavy lifting, not only in anti-inflammatory results, like Dr. Rosario said, but also in the homogeneity and amount of collagen that is generated at 21 days. We have to review this patient again next week, so I'll keep you updated about that. So my conclusions are that really delivering the exosomes on immediately after the treatment improves regeneration, wound healing, neocollagenesis. But if you can't give them immediately, even late delivery exosomes can have a lower but still very good effect. Thank you very much.
Thank you. Thank you for doing that. All right, so we're gonna pass the reins. How about a big hand for all the speakers, and thank you for doing that. And Inigo, you're up. We're gonna go right into part 2 of the lectures. So while Inigo's coming up, you're gonna introduce the group, and we will have a question and answer after this panel. I have it right here.
Well, thank you very much. We're moving to a section where cases are going to be presented. So let me introduce you to Ramon Garcia from Mexico City, Mexico.
I will present the case pediatric oral mucosa and lip tissue regeneration in a Stevens-Johnson syndrome patient. I'm from Mexico and the Children's Hospital of Mexico, Federico Gomez. We declare there is conflict of interest. And it's a really honor to present this case about a 16-year-old Mexican girl who was affected with the oral mucosa and lips with ulcerations and bloody scabs that affected the oral feeding. She was medical history with epilepsy. She was treated with lamotrigine. 1 week before, she was diagnosed with Stevens-Johnson syndrome and confirmed by biopsy. She received treatment, a standard with immunoglobulin and methylprednisolone for 3 days. Despite the treatment, she persists with pain, ulceration, and bloody scabs. For this novel treatment, topical treatment, we use a mixture of exosomes with hyaluronic gel. We obtained the informed consent and ascertainment consent, and the surface area was cleaned with saline solution, and we applied the exosomes and hyaluronic gel in the surface area with sterile swabs twice daily for 3 days. Next day, she was eating. And the 3rd day, this is our result. And this is our result in the 6th day. This is our before and after result. Stevens-Johnson syndrome is an immunological disease, in this case related with medication, that characterized with inflammation, pain, ulceration, and denudated skin. Exosomes and hyaluronic gel treatment was a novel treatment that created a protective barrier maintaining the exosomes and the surface, improved immunomodulation, and accelerating the repair and regeneration, a distinct concept from the traditional wound healing. These are our outcomes that improve the quality of life of the patients. But in the hospital, there is not the first time we are using exosomes. We are using exosomes in biological dressings, and we want to share this experience. This is in pemphigus flaccus. In this case, it's an ulcerated centrofacial hemangioma. Probably, this is the moment we have to innovate in new products and innovate for new medical devices. For example, hyaluronic gel, biological dressings using these exosomes because we need to investment, we need to develop and research. In conclusion, in this case, exosomes in hyaluronic gel treatment was effective and safe for a pediatric patient. Reduce morbidity and enhance the quality of life. However, we need further evidence for effectiveness and cost effectiveness over traditional treatments because probably the future is today. I want to express my gratitude to ExoCoBio, Benev, Auvergne Group for this opportunity and all the health professional work in these cases. Thank you, everyone. 감사합니다. Thank you.
Thank you very much, Ramon. Let me introduce you to Ana Cecilia Amador Ruiz from Mexico City, Mexico. Welcome, Ana.
Hi, good day to all. It's a pleasure to be here with you and having the opportunity of sharing a clinical case with you. I thank my patient for allowing me to share her case. I'm Ana Cecilia Amador Ruiz from Mexico. I'm family doctor, 12 years and master degree in aesthetic medicine. For the past 2 years, I have been studying and working with exosomes, both for medical and aesthetic purpose. In the two scenarios, I have a great result. Allow me to address the dermatitis context. In a clinical dermatology, the term dermatitis is used to describe a variety of different skin disorders that share the same patterns of inflammatory reaction with similar clinical manifestation. This is Danny. Danny is a 30 years old patient with no improvement previous medical history. Start June 2022 with erythema and hands plates on hands, knees, elbows, face, and finally in buttocks. The clinical diagnosis by her dermatology was pityriasis rubra pilaris, such atopic dermatitis, previous treatment with topical steroids with no relevant improvement. This is the result of biopsy of the dermatology. Why I decided to apply exosomes? Well, exosome treatment was based on clinical studies referred to Seung Chon and Irving Duncan articles. These are about the clinical application of exosome in regeneration medicines, anti-aging treatment, and for anti-inflammatory functions. Well, the subcutaneous tissue is the deepest layer of the skin and has fatty tissue and connecting tissue. This layer holds the largest numbers of adipose stem cells in the body, and their exosomes are fundamental for the health and aging of the skin. Anti-inflammatory function inhibit the production of pro-inflammatory cytokines such as TNF-alpha and IL-6, and promote the production of anti-inflammatory cytokines such as IL-10. The treatment history, I used exosome complex, was placed transdermal, 3 sessions, monthly, the first application in September, second application in October, and last application November. This is the first result. The patient come back after one treatment. It's observed 1 month later the significant decrease in the damaged area. And this is the result the before September and the last photo in November 15. The final result, this is taken in August. And the conclusion, the stem cell-derived exosomes can be used to treat incurable of very difficult skin disease like dermatitis, psoriasis, scleroderma, skin fibrosis, and so on base of the dual and synergic function and regeneration and anti-inflammatory. Hansamina.
Thank you very much for this extraordinary result, Anna Cecilia. Let me introduce you to Julia Tvarani O'Shea. She's from Dublin, Ireland. Welcome, Julia.
Thank you, everybody. Thank you, everybody. I would like to do a marathon slide here. Okay. So your hair is your crown and glory, the crown you never take off. So this will be the list of patients I've treated. So yes, you can see the first picture showing that it is a medical doctor actually on 2 oral medication, finasteride and minoxidil, but nothing changed. As you can see, only 1 treatment of exosome gives such glowing hair, healthy scalp, and hair growth. So the next one is, you can see the thinning of the hair overall, but after 1 treatment, very quick, 12 weeks apart. This is a young chap with obviously receding hairline and also a bit of dermatitis at the crown area. Just after 1 treatment, you can see how healthy and fluffy. Obviously, the fiancé is very happy. So this is just showing you the crown area. You can see how healthy the hair at the same place, same lighting, and you can see the color of the hair improve. So this is just to show you the hairline and also the texture of the hair. This patient has psoriasis, as you know, in Dublin, Ireland, Celtic skin, and they are prone to autoimmune and inflammatory skin and scalp condition. So I treated him. This would be 9 weeks post first treatment, and so far he had 2 treatments. You can see even the frontal area, the hairlines has improved. So this is another friend of mine who's a pharmacist. As you can see, overall, improve of hair texture and color as well as hair growth. So this is a lady with psoriasis as well. You can see that after 1 treatment, the hair has grown, and then you can see the texture and the color of the hair improve as well. So just for testing, basically he's my father-in-law. So just trying to see whether the exosome works for his age. He's about almost 70. You can see the thickening of the hair only after 12 weeks from the first treatment, and so far he had 2 treatments. So frontal fibrosing alopecia, I think Dr. Inigo has covered clearly. And so this patient already had all the cocktails given by her dermatologist 10 years ago. So came to me and I decided to do the exosome treatment. As you can see, like only 4 weeks post first treatment, and she's very happy. This shows the quality of the hair. You can see she got her shine back and also her natural curl. And if you focus closely, this is before her 3rd treatment. The slide over there, you can see that baby hair is growing over her actual hairline. So filters are great, but great skin is better. Here we glow with exosome. So as you can see here, rosacea improved. Not only that, you can see like focus over her jawline. There's a bit of lifting effect, and she's very happy just after first treatment. This is another lady with very sensitive skin. I call this microneedling exosome treatment as a red carpet treatment, because you can see that she have a really red face after I done about 1.5 to 2 millimeter depth of microneedling. Look at her the next day, flawless. So this lady has been suffering from acne because she has PCOS, and she was on metformin, and she's getting married soon, so I decided to do the exosome. And also she was given ExoBalm and also cell treat skincare. So only after 1 treatment, as you can see, the scar improved. You know, it's very common in this skin tone group, they can get PIH. So the earlier you treat, the better the outcomes. As you can see, she told me before she started the exosome treatment, she decided to stop her metformin. And guess what? Her LH FSH ratio has normalized. So I think Byung Cho should investigate further. It could have, it could be the effect to the body, the exosome. So I just skip this. This is myself. I developed atopic dermatitis. I've tried all the topical treatment, including tacrolimus. no effect, and after one session of microneedling with exosome. Amazing. So no high-tech treatment, no fuss. All I need is the magic cream, which is the ExoBalm. So this will be the list of patients I treated. So this patient has melasma and also rosacea. So I need to treat the rosacea first before I move on to melasma. As you can see, 1 week after ExoBalm. So the last case will be dermatomyositis. This patient has been suffering from a skin condition for years and only diagnosed 3 years ago by one of the dermatology. And as usual, they treat with steroid, first-line cocktails, but nothing really improved. Look at him, just using ExoBalm 2 weeks later. Happy man, happy wife. So, in this case, I couldn't do microneedling because there's so many broken skin, but look at him at 4 weeks. I think his skin looks better than mine. I'm very happy, and he's continued to improve. So I actually maintained him with the firming cream and also the other cell treat product. So this is the wife. So a short message.
Appreciate.
What was the message?
Sorry.
Of the wife?
Sorry. Yeah.
What did she say?
Look, just to say thank you. We appreciate what you sent us. And I suppose there's more appreciation for the skin as it has improved so much. I mean, we're years and years going to dermatologists, and nothing as recently as about 2 months ago, and nothing could be known that he was happy with ever. So thanks again.
Thank you everyone for your
Thank you very
attention.
much.
Have a lovely evening.
Thank you very much, Julia. It's interesting you noted a change in the pigmentation of hairs after the treatment. So let me introduce now to Dr. Ali Alamri from the Kingdom of Saudi Arabia. Welcome, Dr. Alamri.
Thank you so much. They informed me that the time is short, so I try to pass over all these slides. So actually, I'll not go through all slides, but okay. It's too late. Yes. So understanding the pathophysiology, and I find this the gold standard to get the optimal result. So I'll go down to the clinical experience. So we find some protocols in our background. So when we mix the ASCII with PRP or ASCII with Regeneron, we find a good or excellent result in the androgenic alopecia. So also in skin rejuvenation and scars, when we mix the ASCII with drug delivery system by radiofrequency microneedling or CO2 laser, we find also the best result in our cases. ASCII phase plus hyaluronic acid or tranexamic acid also work excellent with the pigmentation and post-inflammatory hyperpigmentation. Actually, we apply also the ASCII in preocular dark circle and in cosmetic gynecology. I find the best word in the use of ASCII in skin texture is regeneration instead of rejuvenation. We find some protocols to distribute the ASCII over some micro-injected points in distance in every 1 month and about 2.5 ml per session, and we find improvement in the skin texture, as you see here, the fine wrinkles. In the next case, you'll find also improvement in around the dark circle eyes and also nasolabial fold, as you see here in this case. What you will find if you inject the micro points of the ASCII, you will find in the first week a good hydration, and after 1 month you will reduce wrinkles and pores, and after 1 month you will find increase in volume also and skin texture. My protocol in around the eyes, I mix 1 ml of ASCII with 1 ml normal saline or hyaluronic acid, and I find excellent result after 3 sessions. So as you find here, before and after. And also this case is actually a difficult case to treat with hyaluronic acid alone as filler, and I find the excellent protocol as I mentioned before. Androgenic alopecia, you know it's a challenge to treat with topical, so this case I mix the exosome with Regenera, and the result, as you see, after 1 session, good improvement. This case, I mixed the ASCII with PRP, Magellan, and this is after 2 sessions. As you see here, I only use ASCII injection, micro injection, and the result after 1 session. As you see here, after 2 sessions, and here after 2 sessions, here after 3 sessions, you see improvement. We change also the texture and color. As you see here, the big improvement between the 2 cases, and this case is telogen effluvium, and the result after 3 sessions. This is also androgenic alopecia for female after 1 session. I will pass over another indication, surgical and acne scar. I think we have to use the drug delivery system, as my colleague mentioned before, to use the CO2 laser or radiofrequency. As you see here, the drug can go deeper and get more benefit. So in this case for acne scar, I did CO2 laser and I apply only as film of ASCII, and the result after 2 weeks. And this case again, I have no more time, so I have to pass over this patient after trauma, and this is after 2 sessions. We can find improvement for hyperpigmentation also to apply the ASCII with radiofrequency. This also brachoplasty scar, and this is after 2 sessions. This one also after 4 sessions, and this one after 4 sessions. And here, as you see, the first session and the second session. This is also another case. Here, inflammatory disease, actually I find excellent result with the lichen planus in the scalp after 1 session. Here, alopecia areata, you will see a video for this patient has allergy against the prednisolone and an steroid, so I inject him alone with ASCII. This is the improvement after 4 sessions. This is active acne, and I use the microneedling radiofrequency with ASCII as film. And this case also the same. Hypertrophic scar and burn, I find this excellent result also because we try to avoid the hypopigmentation and atrophy after injection of the steroids. So I mix the Conalog with the ASCII, and you see the result in the other side. This case is baby with burn, and I apply the ASCII, and this is the result after 3 days, only 3 days. This is also old burn scar, and I treat her with ASCII and CO2 laser. Dark circle, again. And this is the conclusion, the combination with other treatment, I find it the optimal treatment. Thank you so much.
Thank you very much, Ali. I mean, they are amazing cases. We all learn from this. I will try in the same indications. Thank you very much. Are you from Jeddah or Riyadh?
Riyadh.
Riyadh, okay.
Yeah.
I've been there several times. Well, thank you very much. Let me introduce now to Dr. Victoria Bello. Victoria, please welcome to the podium from the Philippines. Victoria has 13 big clinics in Manila, right? 12 of them are in Manila. And I've seen their organization, and I was blown by the efficacy and the efficiency of 600 people you have working for you, and with amazing results. So please, Victoria.
Thank you, Dr. Inigo, and thank you, Dr. Michael Gold. These two are my mentors. They've taught me a lot. Our clinic has been around for 33 years. I started when I was 10 years old. You can tell. Now, anyway, we have 60 doctors with us and 600 people. So we have 3 cases to present. These that were done by our doctors. This is great. Okay, here we go. Next. Oops, sorry. Here I'm getting. So our first case is a combination treatment of exosomes and a 589 vascular laser for the treatment of psoriasis vulgaris. So she's a 35-year-old female. She has a known case of psoriasis vulgaris since she was in her childhood. There were no treatments. You know, she tried a few psoriatic treatments, but she thought they weren't working. So she only puts argan oil to soothe the itchiness and the scaling. And then she came to our clinic and we applied. We did 2 sessions of the Advalite, the 589 laser, a vascular 1,000 shots, and then photos were taken when we started before and 1 week after the second session. The sessions were 2 weeks apart. The patient tolerated the treatment very well and no side effects were noted. So here we have a picture of the right leg and the left leg with the psoriasis. And here he is after 2 sessions of 2 Advalite treatments and the application of exosomes after every treatment. So there were 2 applications of exosomes, ASC, exosomes. Conclusion, treatment combination of exosomes and a vascular laser reduced redness, itchiness, and scaling of psoriasis vulgaris. Next slide. ExoBalm, the second case is, so the first case was by Dr. Jack Luna. The second case is by Guada Capis, I think. So ExoBalm is monotherapy for non-healing post-surgical wound treat due to hidradenitis suppurativa. So we have here a 24-year-old female who was diagnosed with hidradenitis suppurativa 4 years before coming to us. She had multiple recurrent painful nodules, abscesses, and draining sinus tracts on both armpits, inguinal areas, and the nape of the neck. On September 2022, the patient underwent surgical excision of the ulcerated sinus tracts on her right arm, because that was the most malodorous and the most serious one. And then it was left to heal by secondary intention. Topical antiseptics and antibiotics were applied as well as oral antibiotics. But the wound remained open for 8 months. And this is when she sought treatment at Bello with Dr. Jack Luna. So week 1, here we are before applying the exosomes. It was ExoBalm. So we only got, we had very little ExoBalm at that time. She only got 1 jar, which is supposed to be finished in about 2 weeks, but she used it for 7 weeks. Very little, she really tried to save the balm for 7 weeks. So here we are at week 1, and at week 3, you can see already there's some closure of the wound. And at week 6, it's shrinking. At week 7, here we are, it's almost totally closed. And here she is at her right armpit. So 8 months of an open wound, in 7 weeks it closed. So the conclusion would be that monotherapy treatment, so there's no antibiotics or anything, with ExoBalm was effective in the treatment of non-healing wounds. The third case is the management of radiation dermatitis in a breast cancer patient with ExoBalm. So we have here a 48-year-old female diagnosed with invasive breast cancer on the right side. She went under lumpectomy on June 2023, and then she did radiation every day for 30 days. That resulted in a lot of blistering and post-inflammatory hyperpigmentation. ExoBalm was started after her 30th day of radiation. Past medical history, she underwent the same treatment in 2015, where she also underwent 30 days of radiation, and it took 3 months of emollients for her post-inflammatory hyperpigmentation to resolve, but the scar was still thick. So this time we started ExoBalm the day after. And here we are on day 1. As you can see, there's thickening around the nipple of the scar and very marked post-inflammatory hyperpigmentation. At day 20, after applying ExoBalm, you can see that the PIH is much better and the scar is much flatter. And this is day 35 follow-up, and you can see resolution. A lot of the hyperpigmentation has disappeared, and the scar is much flatter. So the conclusion is almost complete resolution of hyperpigmentation and improvement of thickened skin and surgical scars. So I'd just like to thank ExoCoBio and Beyond for introducing us to this product. In 33 years, I think there have been things that have revolutionized my practice, and this is definitely one of the biggest things that has happened to us. And I am so excited to come here every year to see even more uses for ExoCoBio. Kamsahamnida. Thank you.
Thank you very much, Victoria. Amazing cases. Because we're running out of time, we're going to reduce the 20-minute time that we had for questions and answers to 5 minutes. Can you bring 4 chairs, please? And I would like to invite Jan, Michael, Renato, and Rosario. Please come up here to the stage so that we can... is there anyone in the audience wanting to make a question? There's a microphone there in the center. Take this opportunity because we won't have much time for questions. Is there anyone wanting to make a question? Yes. Please, can you tell us your name and who you want to make a question
Hello.
for?
Can you hear me?
Hola.
Yeah, hi, I'm Dr. Nami from Lebanon. I'm a dermatologist. And first, I would like to ask about the half-life of reconstituted exosomes, because I would like to ask a further question once I know the answer. I don't know whom to address this question to.
The half-life of reconstituted, maybe Beyond wants to answer that question.
Half-life of reconstituted exosomes.
Okay, thank you for your question. After reconstitution, our official recommendation is immediate use, less than 3 hours. Yeah.
Okay, in that case, I would like to ask Dr. Rosario, how did she reconstitute all the vials for her patient and send him home with it to use for a period of 7 days? Or
Yes, that's
10
very
days?
easy. There is a diluent that accompanies the exosome, so I just reconstituted it in front of the patient and the family or the accompanying person. And then I applied it, showed them how to do it. And I have in my clinic, because we don't have ExoBalm readily available, I have in my clinic a lot of insulated bags and ice. And I sent it there with strict instructions to put it in the ref and then come back within a week.
Yeah, but the answer was the exosomes lose their
Correct.
capability after like 1 hour or so.
Correct. However, I am following what Dr. Duncan did for her patient. And so based on her experience, I just followed that because my country is very poor and we try to do what we can.
Well,
and it
what I mentioned is that to keep the best potency, 100% potency is 3 hours, we recommend. But definitely after that, slowly, some molecules will be degraded, right? So 3 hours is the best
Is
potency.
the optimal time. So also I have one more question. What is the constituent of ExoBalm and ExoMask? And would you recommend that after a procedure, or would you recommend the reconstituted vials? Which is more potent after like microneedling or whatever? If we have both in our country, what would you recommend? Thank you. That's all I have.
Thank you. In your clinic, for example, post-laser treatment, In Ukraine, we can recommend the reconstitution, ASE Plus. It's the flagship product, but the ExoBalm you can apply once at your clinic, and your patients are supposed to take them to their home for up to 3 weeks.
Yeah, but what do they contain? What is like the number of exosomes in the balm?
The number of exosomes is the same between the two products. The particle number is the same, but the formulation is very different, actually.
Thank you.
Thank you.
Thank you.
We're going to ask one question per person, so we can have more diversity in the questions, please.
Anyone would like to make any other question? Can I make a comment? Because ExoBalm is also reconstituted, and I think that the indication is 1 month. Is that correct?
1 month.
It's up to 3 weeks now. Yeah. After reconstitution, you keep in the refrigerator for 3 weeks.
Okay, please.
Hello, Jackie Lewis from London, plastic surgeon. Can you please tell me if the ExoBalm contains RAES-derived exosomes or human-derived exosomes? Thanks.
You have two options, actually. You have two versions. So it's about regulatory issues. So in your country, RAES stem cell exosomes, actually. But in the US, human stem cell exosomes.
All right. Any other questions?
Right.
Yes.
I'm Dr. Lin from Taiwan. I have an interesting question because we learn the repigmentation by exosome, but we also know that the exosome can treat melasma. Is there any conflict between these two concepts, repigmentation and depigmentation?
That's a very good question. Well, I had the same question. I made the same question to myself. How can exosomes reduce hyperpigmentation and treat melasma, and at the same time can induce pigmentation or recovery? So the fact is that both things happen. So there must be two different mechanisms molecularly, right, to explain that. And in fact, I have an idea. I think that the way we can recover the pigment of a hair is through that pathway that I comment, the programmed cell death, right? And at the same time, if we manage to reduce the pigmentation of melasma, it is because of some microRNA that is inside. So I remember that these days, studying about this, keratinocytes have also contained microRNAs that enhance pigmentation and that reduce pigmentation. The same exosomes of a keratinocyte. I'm not talking about exosomes of adipocyte stem cell derived. So both things happen at the same time, very probably because the mechanism is different. And these are facts. We don't know the real answer. I'm not a molecular biologist like Jean-Pierre, but I think that we've seen a reduction in pigmentation with melasma. We've seen a recovery of color in hairs.
Thank you so much.
Thank you. So anyone would like to make any comment about this?
I mean, one of the things we know whenever you take an energy-based fractional device, whether it's CO2, non-ablative, microneedling is really a fractional device of some kind. Those devices by themselves cause repigmentation in hypopigmented areas, and they also cause reduced pigment in hyperpigmented areas. And so again, I think to me, the exosomes are just eating both of those processes because again, of the wound healing cascade that we know that this plays a role in every part of. So again, I don't think any of us understand all the mechanisms because we surely don't understand it, why in fractional lasers, hypopigmented pigment and hyperpigmented go back to normal skin color. So we just have to keep learning and keep studying.
Right. So I'm Shazia Ali from Jeddah, Saudi Arabia. So I hear that over 1 million cases have been treated with the exosomes. It's great science. We have some amazing cases that we've seen. I was just wondering that are there any reported side effects out of all of those 1 million cases? Because I know this is amazing and we are looking at all the good stuff. Let's look at the other side of the coin also. Are there any reported side effects in the million cases?
So, I'm not aware of any with these exosomes? I'm aware of lots of infections and other things from some other plant-derived exosomes in the United States. I'm not aware of any. And have you had any? So again, I think it's if it's a drug, there's ways to monitor side effects. We have to report those, right?
Yeah.
As clinicians and companies if they get in. So I'm not sure that is the same case here, at least in our world. So have you had any reported?
Before I answer, do you have any comments on the side effects? Bello or Cherry, can you share any experience on side effects?
Sure. This is the one that I reported a while back, that some of our patients in the Philippines experienced some stinging upon the application of ASCE, although the majority of the patients will experience soothing relief from almost instantly, within 20 minutes. So these are a few random cases. So we reported them. And I also reported that in some of our patients, we had transient cervical lymphadenopathy that was seen after microneedling of the scalp. So these are basically some of them. And then there was last night, I attended a webinar by Sci Asia, and there was a physician who reported that he had seen the granuloma formation that happened after the Benev product was injected into an area. Sir?
Actually, I don't think it's true, but from our almost 1 million cases, when we applied our product topically or in a very minimal invasive way or in combination with the energy-based device, there was no report, something like that. But some doctors sometimes inject, I believe, and then they could see some allergies or something like that. But one thing is that exosomes themselves are, I believe that they are very safe. They came from our stem cells. Always, our cells are releasing exosomes in our bloodstream, and they are exchanging information. So exosomes themselves are very safe, but sometimes maybe other ingredients might cause allergy. Just like normal skin boosters, like NCTF, something like that. HA sometimes cause allergy. That kind of allergy could be seen by using our finished products.
Yeah, it's very interesting. I think the journey has just started, and we are going to see both sides of the coin as we move forward, but it's super exciting. Another just question, because while we were touring the factory, they were telling us that we are maintaining a cold chain and we are keeping the exosomes at a certain temperature. And now that we are combining with energy-based devices, so the skin temperature might be raised when you're doing fractionated CO2 or BMYAG or any of those devices, or RF for that matter. So when you're applying them to the heated skin, do we lose some of the exosomes while we see some of the results which are from the remaining ones, but do we lose some product in the process because of the heat? Any ideas?
Yeah, in our acne scar publication, which is a double-blind one, after laser treatment, they waited for about 20 minutes to cool down. It must be like that to be more effective, I think.
Okay, we're going to take 3 more questions.
Just a very quick comment there. I think if the skin takes the heat, exosomes are part of the skin and the tissue, then the vesicles from the product should also be able to take the heat. That's just my guess.
It'd be interesting to know, though, exactly what happens.
Thank you.
Dr. Subaru.
I'm Suparut from Bangkok, Thailand. I would like to ask Mr. Cho some of the questions, but don't be angry with me. Okay, first you mentioned that ACE should be used for 100% efficacy within 3 hours, right? But you mentioned that ExoBalm at the same source of the exosome can last for 21 days, 3 weeks. So if we store the exosome in the refrigerator and keep the control of the temperature, so we can use it for 21 days, like ExoBalm, right?
Thank you. Thank you for your question. And, so ExoBalm is a kind of a different formulation. It's cream type formulation. So it's, maybe it may lose more potency than ASE because it should be kept. But our recommendation is a week. But people, doctors and patients want to use ExoBalm longer, in a longer time. So, and the amount of the cream would be like, can be used for 3 weeks. But our official recommendation is fast use, actually, because they might lose their potency in the refrigerator while being kept in the refrigerator.
Would you share the information if we store the ASE exosome more for 24 weeks? What is the potency of them will decrease from 100?
Okay, we have internal data. The data came from one of our professors about 4 years ago. I will share the data with you later.
Okay, thank you. Next question, please.
Hi, good afternoon to everyone. I'm Dr. Suzanne Twin from the Philippines. I have 2 questions for Dr. Latval or anybody who can answer. The first is, because of all the products available in the market now, number 1, is there a minimum number of particles for the exosome product to work? Maybe 2 billion, 5 billion, 10 billion. And number 2, is there a saturation point where after putting so much exosomes, the results maybe begin to plateau or, you won't see any results anymore after that?
So I'll answer the second question first. There's always a plateau of pharmacology, right, where you stop seeing additional effects. It's basically you have a dose response curve and then you don't see an additional effect. So that would also be true for exosomes, I'm sure. The other question was regarding the number of particles. Now this is really very important. Particles doesn't mean exosomes. Particle means there's something counted by a nano-tracking analysis device that is approximately up to 200 nanometers large in the solution, in the preparation. That doesn't mean that those are exosomes. Yeah. So if you, for example, you mentioned plant products, if you take mango or something and put it in a mixer and create nanovesicles out of that, there's a lot of other stuff that is present in that exosome isolate. So you shouldn't say that you need 5 times 10 to the 9, 5 billion, or 20 billion, or 8 billion. Depends on the quality of the vesicle.
So because there are different sources now, of course we have adipose tissue, we have bone marrow, umbilical cord. Do the number of exosome particles matter?
The quality is much more important than the numbers that's on the bottle.
All right.
Quality is everything, actually.
So I get what you're saying, because there are companies out there that, you may have 5 million and they have 8 million, and somebody I know says they have 20 million and billion. And what do you need 20 billion, right? If the quality is not right. And if you have 20 billion irregular particles or or not the right quality, we as the clinicians have really no way to figure this out yet. So we rely on you guys to help us.
So what's probably needed is, I'm not in the business of comparing different products and their quality and their functionality, but that should probably be done in one way or another. It's probably difficult if one manufacturer compares all the different products, probably better than somebody independent does, right? So some sort of functional anti-inflammatory effect would be good to compare, I think.
Okay. Thank you,
Well,
doctors.
thank you very much. We are running 35 minutes late. So may I suggest, if there's any more questions, please, I would encourage you to make the questions directly to the speakers. Thank you very much for this panel. It has been great. And we can restart 10 minutes to 7, all right? At 18:50, okay? See you.
We're actually going to have 45 minutes, I'm sorry, and I appreciate your patience today. So we're going to have 45 minutes for dinner. It's going to be here inside of the room, so if you need to go to the toilet, you can quickly go and come back. And there you're going to have your dinner served in your tables. All right, so we're about to start this third session, so I'd like to call our moderator for tonight, Dr. Fernando Macedo from Brazil. Please welcome Dr. Fernando Macedo with a big round of applause. So we're gonna have third and fourth sessions without any break, so please be patient. If there's coffee outside, if you wanna drink, just be quiet. And then welcome to Dr. Fernando Macedo.
Okay, hello. They told me to introduce myself, so I am a Brazilian dermatologist, and I have been working with lasers and aesthetic medicine for 25 years. I had a fellow in Hospital Saint Louis in Paris in 1998. Some people were being born at that time. And I love what I do, and exosomes is something that really caught me when I saw in the first Congress in Europe and in the United States. It was something that would, make my work maybe better, and for my patients also. I live in São Paulo. I'm launching my third clinic there. I have 30 dermatologist assistants, not just dermatologists, surgeries and gynecologists working with me. And I think, since you probably know that Brazil is the second second market in the world, we're very excited about bringing exosomes to Brazil, to a great dermatologic surgery society and a great market also. So I learned a lot here now, and I've been learning from exosomes. I know some of the speakers already. So it's an honor to be here with you and to share my learnings afterwards with my colleagues in Brazil. Also, there are some other dermatologists from Brazil that I would like to thank them to come also to Korea. So, having said that, we're gonna introduce our first lecturer. Is Mr. Tom Siri. He's the CEO of Real Self, and he's gonna talk about Real Self in medical aesthetic industry. Should be a very interesting talk.
Thank you. Oh, there we go. Thank you so much. Thank you for the introduction. And I'm very accustomed to eating. I have 2 children, and when they're eating meals, they barely pay attention to me. So if you continue to clank and use your dishes, fine. I have no problem. I do appreciate being here. I'm not a physician. I've never run a practice, nor should I probably ever be running a practice. I'm an entrepreneur. And what has attracted me to this industry, I know many of the doctors in this room. I've been over 15 years focused on medical aesthetics from a digital perspective, digital marketing, is that so many of the doctors that you heard today, like Dr. Saltz and others speak to, they're entrepreneurs in themselves, and many of you are in this room. Putting yourself out there, looking for a problem to solve, using your passion, your grit to really face the struggle that entrepreneurship really embodies. Because that's the difference between an idea and entrepreneurship is actual execution. Included within there, of course, are the industry players that, like our hosts are, and Ethan, just incredible entrepreneurs who are willing to push the boundaries and find a product market fit against all odds. So I'm honored to be here, and I'm going to speak today to a bit of the insights that I might be able to impart from my position in the industry, from more of a looking at consumers, looking at past trends, current trends, and see where regenerative aesthetics is headed from a different angle, perhaps. One thing that was poorly vetted about me is that this is a talk I gave in Las Vegas not too far in the past. And you can see I totally knocked out this guy. So if you feel a little sleepy, there's some people here I know are suffering some jet lag. I would not take offense. It's happened to me before, and I work on Powerful. The platform that I built over 15 years ago was really centered around helping consumers become more confident, more educated about aesthetics. At the time, there was no Facebook really, the iPhone hadn't been invented. It was a very uncharted space, and it was very murky. And my wife had said she was getting a facial, and they encouraged her to get a laser treatment. And she said, I was like, oh, I didn't know you could do a laser on your face to look prettier. That's very odd, to burn your face, look prettier. But she said, yeah, and everything all they gave me was this brochure, and I don't really believe anything in it. And at the time I was working for Expedia, we had bought TripAdvisor, and we were just talking about this. And TripAdvisor had really revolutionized this idea that you're not just taking what marketers say, what the hotel says about themselves, you're seeing what real humans are saying. So I applied that same thinking to medical aesthetics, and we've built a platform that's evolving to international. We have Middle Eastern presence. We're across 15 countries and many languages. And I'm very proud of our team and the spirit they have. But we're also going through a revolution, or sort of, I guess, disrupting ourselves before someone potentially will disrupt us, which is we're using AI now to figure out how we take our over 4 million Q&As that many doctors in this room have participated in, and I appreciate that, and train language models so they can be more effective at answering consumer questions. And it's a very challenging space, ChatGPT, ChatGPT-4. In medical, it's actually, and I'll just digress a moment, AI is complex in that what we've seen is the more you interact with an agent, when you're chatting, the more it hallucinates, the more information it makes up. So it's a very, if you think about your own patients, for you who are providers, doctors, they have more than one question. They have immense numbers. So that's a space that we're tackling. It's gritty, it's difficult, but it needs to happen because that's something we'll talk about, the trends. One of the things that Ethan challenged me with is regenerative aesthetics a thing? What do we see? At the center of anti-aging medicine with aesthetics, what can we glean from my perspective? So it's an exciting space because I do think there's some very favorable trends. I'm not really here to make predictions. Really, I think most of us are trying to figure out, and this is using ChatGPT's definitions, we really are trying to seek out what trends are there. And as they say in many things, including investment, the trend is your friend. You want the trend behind you, or you want to attach yourself to moments where things are changing favorably. And the trend analysis involves insights that come from very unique places. For instance, Nike will put people in Seoul, Korea, just to look at the fashions and to see what people are wearing, to learn how to inform designers. You see that across the board in industries. There's this insight gathering that's really sifting through and trying to build awareness and direction. Not lost in this room is kind of almost redundant to say medical aesthetics is a lifestyle trend. And I think in what I've been just blown away with in Korea is just how it's so much further ahead of the US. It's so advanced. This is of course it's been a trend. It's been a trend for a long time. But really we saw with COVID in the United States, this acceleration where consumers are searching and finding, and I use Botox as sort of almost a leading indicator of how common it is for consumers to now consider that as part of their lifestyle. It's not just a thing for the rich. It's not just a thing that people kind of hide. It's become a very much normalized and comfortable where people share on social media and so forth. One of the things that's kind of unusual about my perspective on this, I think, is that it's not just the trend of medical aesthetics, but what's happening in technology. And this is a chart from Thomas Friedman's excellent book, Thank You for Being Late. And, this is basically showing on the, our ability to adapt, our ability to process information is relatively stable. We evolve, but it's a slow process. But with quantum computing, and we talked about AI a moment ago, you see it's exponential. So at this point, we have, for instance, AI technology that enables the software test for becoming an Amazon engineer was aced by ChatGPT. Aced, 100% right, all the answers. Now there's some flaws and so forth, but the point is what science, what technology is going to enable is there's going to be some challenges. There's going to be some very difficult societal challenges we are just on the cusp of. That's including disruption of entire fields where jobs will be lost and replaced by bots and other technology-driven solutions. But what it does do, it should free a lot of us from the toil, from the difficulties that many of us have with everyday things like booking a restaurant. Very simple things to more complex, like finding compounds, your next compound in drug discovery. What we see in US consumers, and I don't think this is different for many places in the world, is it particularly a pronounced demand, not just desire, but demand for greater convenience. They're willing to sacrifice other things as long as things become more convenient. In America, I typically do a little thing where I ask the audience how long, we use Uber quite a bit for cabs. I'm like, how long are you willing to wait? And the typical answer is about 4 minutes. And I always say that's insane, but that's your patient, that's your customer, that is the generation, that's how we see things now. They deliver to us, they should happen instantly. And these are trends that do play out in medical aesthetics and to regenerative aesthetics as well. I'll share that in a moment. What we see from our perspective, when I started my business, things were a little simpler. You could map out the journey, the consumer, customer, patient journey, where, oh, here's what they do. And it's sort of somewhat linear. It wasn't that hard to be a good marketer in the early days of the internet, as well as in aesthetics if you wanted to sort of become more successful through digital marketing. But what we've seen is, and this isn't just medical aesthetics, it's hyper complex. If you go to a marketing conference, they just spend the entire time talking about how this is impossible to figure out, how consumers make these decisions. And when you ask your patients, where did you hear about us? And it just could be like 40 different things. How did you hear about exosomes? I don't know. It just came up in a conversation. I saw it on digital. I said, it's hyper fragmented, which just means it's more and more challenging for us to actually know which node we spend time on. Where do we make it? Where is the most influential places? Where can we have impact and how do we affect it? But this is how we all operate, not just your patients, not just industry. Where we see it playing out in our data is that consumers have, particularly these new wave of mainstream consumers. And again, you're aware of this, but I think the rest of the world is sort of slower to catch up, is that they are extraordinarily focused on how do I get results conveniently delivered so I can go in no downtime. This whole Zoom boom where you can hide behind a camera is over now. And so what's next is convenience, going back to convenience, simplicity, results. Actually, we see in our own data, the second largest topic on our platform last month was Morpheus 8. And I have no relationship with the company, but it's just an indicator of just how big that interest is. And it's not about Morpheus 8. It's about what they think it delivers, the benefit that consumers are seeking. Our own editorial staff has been looking closer at exosomes, and they share a lot of what you've heard today from their editorials, sort of sifting through, talking to beauty industry people, and have said there is something here because exosomes play that role and could be a very important part to delivering what that consumer now demands. Simplicity, back to work, results, simple, making, and gosh, I'm going to have to go to, in Barcelona and get my hair work done because I like the results I saw. Dr. Gold, I'll talk to you later about that. But again, we see it as something that's interesting and what could be next in the industry, which is something that's really exciting, particularly for this room. But this is my last slide. Sorry, I'm at 30 seconds. But what stands in the way? And this is more of me putting it on you, saying education. You can't do enough educating. The lectures you heard today, and we'll hear this evening, the insights that you have. Consumers don't just want to hear, at least from what I've seen, now it could be different in Korea, they don't just want to hear what that brochure says. They want to read the papers. They want science. They want to learn. But look at the gaps you see in what US and UK consumers have about technologies that you've all taken probably for granted that, of course, everybody's heard of IPO. IPO's been around forever. So there's a tremendous opportunity to play a role in this and helping the consumer get educated, help them understand what these new technologies do for them and deliver what they ultimately want, which is that convenience factor and knowledge that they're making wise decisions and they're really going to achieve their confident self. So with that, I'll say thank you so much, and I really appreciate it. And if you want to reach out to me, please do.
Thank you very much, Tom, for your insights. And now I have the pleasure to invite Dr. Sailin. She's one of the researchers that worked together with ExoCoBio from the beginning. She's also called the mother of the exosomes, I think. And it's a great pleasure. I have seen her lectures before to have her here, and she's gonna talk about anti-aging effects of mesenchymal cells exosomes from Singapore. Thank you.
Thank you very much. First, I'd like to thank ExoCoBio and CEO Cho for this kind invitation to be here. Every time I come here, I see great progress in ExoCoBio. It's very impressive. So I'd like to start and share with you another aspect of MSC exosomes is anti-aging effect. Now, so I think most of you are familiar with MSC. It's been used to treat many diseases many years ago. And for a long time, it was thought that they were home to injured tissue and graft and differentiate to form new tissues. However, in 2005, the scientific community started to realize it doesn't work that way, and they think it works through the secretion. And then, by 2009, 2010, the field discovered extracellular vesicles, first by Kamusi, who reported microvesicles, and a few months later, my group reported that the exosomes, the smaller vesicles, are also active. Now, I'd like to share with you how we moved from cells to EVs. So initially it was Kamusi group reported that it was the 80 nanometer to 1 micron microvesicles. Then we say that the 100 to 200 nanometer exosome also works. And by 2017, the Kamusi group observed that the 160 nanometer, but not the 200 nanometer vesicles are therapeutic. So right now, the general consensus is that the therapeutic MSC EVs are between 50 to 200 nanometer. So I'd like to share with you how I got into the field. So we were playing with human ES cells, and we got MSC, and we found that this MSC could produce conditioned media that is therapeutically active, and it could reduce the influx size of animal models by about 50%. But the conditioned media of HEK cells do not. Furthermore, we found that the active agent in this conditioned media reside in the less than 200 nanometer range, that is less than 1,000 kilodalton. And then we subsequently isolated exosomes, which could do the same thing as MSC itself. And we also, in about 2011, most of us are aware that MSC are very heterogeneous, they have a finite lifespan, and that introduced a lot of variability in the manufacturing process. So we decided then to clone the cells, immortalize it, clone it, so that we have identical cells for infinite production of MSC exosomes. And with this immortalization, of course, there there were concerns that it could be tumorigenic or produce exosomes that are tumor promoting, and we've shown that it does not. And then using these exosomes, we found that it could be efficacious in many diseases. We and various collaborative groups around the world show that it's effective in more than 10 different diseases. And last year, we carried out a phase 1 clinical trial on these exosomes for topical application to treat psoriasis. Now, from 2010, when Kamusi and I published the first paper on MSC EVs, there's been an exponential increase in publication on stem cell EVs. Last year, there was a paper that surveyed the stem cell EV research around the world. As you can see, the powerhouse are US and China. They dominate the R&D in every parameters you can think of, except one. That is the average citation per paper. In this case, Singapore was number 1. We have more citation. We have twice as many citations as the US and 3 times that of China. And I like to think that my postdoc and I had something to do with that. So today I would like to talk about the aging-associated senescence that these MSC exosomes could have. So it has a lot of implication in anti-aging. Now this work was done and published 2 years ago. It was done mainly by Paul Robbins, who led this study. So he uses the mouse model of progeria. For those who are not familiar, progeria is a disease of premature aging. So most kids die in their teens. Now, so one of the first experiments that Paul did was to inject MSC into these progeria mice. And then what he observed was that at 60 days, 90% of the mice were dead, untreated mice were dead. But 90% of those that were given the young MSC were still alive. Then he injected these animals with MSC EVs. And what he observed is that by 60 days, 100% of the untreated mice were dead. 60% of those treated animals were still alive. So, as you know, in aging, right, most of the time, it's now thought that the aging process is caused by the accumulation of senescent cells. And this accumulation causes a lot of age-related disease. So a lot of the anti-senescence therapy or anti-aging therapy target these senescent cells. So a lot of drugs are being developed to remove the senescent cells, called senolytics, or to change the phenotype of the senescent cells, called xenomorphics. So the question that Paul asked was, does these EVs change the senescent cells? So he put the EVs on these cells, and he looks at one of the characteristics of senescent cells. So senescent cells has many characteristics. The easiest one to measure is the beta-gal activity. So he looks at beta-gal activity, and he shows that the EVs can reduce the number of senescent cells that is senolytic. However, then he ran into a bit of a technical problem. So because they couldn't produce enough MSC exosome for their study, so he decided to collaborate with me to use the exosome from our immortalized cell. So when he take these MSC exosomes, he used it to treat his mice. And then he looked for aging symptoms. Can he reduce the aging symptoms in these mice? And what he observed is that after treatment with this MSC, he can see a significant reduction in aging symptoms in these mice. Then, so he can show that these immortalized MSC exosomes reduce aging symptoms in the progeria mice. The next question is, what aspect of the senescent cells is he reducing? So he took the cells, the exosomes, he put them on senescent fibroblasts. And he looks at some of the characteristics of senescent cells and asks which can it reduce those senescent markers. The first one he looked was beta-gal activity. So he saw that the beta-gal activity was reduced. Then he looked at the senescence-associated secretory phenotypes. And these are the IL-1 beta and the IL-6. And he also observed that both cytokines were reduced. Then he looked at the cell cycle arrest. So senescent cells are arrested in the cell cycle, and these are the markers for this is P16 and P21. And he also showed that the exosomes can reduce this senescent marker. So the next question was, he wants to know, so he has been working on these progeria mice. These are genetically defective mice. So he wants to know, can he see the same effect in naturally aged mice? So he take 2-year-old wild-type mice, which is very old, and then he inject them with the EVs. And after he inject them, on day 0 and day 7 and on day 10, he sacrificed them. He removed their liver, the kidney, liver, lung, and brain. And he looks for senescence marker. So these are the senescence markers. So the first one he looked at was a cell cycle arrest. So you can see that in the kidney, liver, the P16 was reduced, but not in the lung and the brain. And in the lung and brain, the P21 was reduced. Essentially, one of the cell cycle marker was reduced in at least one of the marker is reduced in all 4 tissues, which means that the cell cycle arrest was not cell cycle arrest. Now, the next one he looked at was the senescence-associated secretory phenotype. And what he observed was that in all the tissues, except for the lung, the IL-6 and the IL-1 beta and IL-6 was significantly reduced. The IL-6 in the lung was not. And this is a lot more important because it's generally thought that this secretory phenotype is the driver of the disease phenotype. So decreasing it will reduce the disease phenotype. So I hope this is something informative. I'll conclude now. So basically, I hope that I can convince you that the immortalized MSC exosomes could reduce cellular senescence both in vitro and in vivo. And therefore, it could potentially alleviate senescence-associated phenotype in human, this is the target of most of the anti-aging strategy at this moment. So with this, I'd like to thank you for your attention.
Thank you, Dr. Lin, for a very interesting talk. And now our next speaker is Dr. Richard Jim from the US. He has been helping develop exosomes in the States and was one of the first person also who I talk about exosomes. I thank you for your time. And he's gonna talk about utilizing the power of exosomes to treat alopecia.
All right, well, thank you very much. And thank you, Byung Cho and Ethan Min, for giving me this opportunity to speak in front of you. I just want to talk a little bit today about how I've used exosomes in my practice in treating alopecia, some very practical approaches, also elucidate a little bit about PRP and how well that's worked, but also show you a comparison of exosomes from platelets and exosomes from adipose-derived mesenchymal stem cells, and showing you some fundamental differences. So hair loss, this is just general statistics. Bottom line is both men and women were affected, right? When we get older, hair thins, we lose hair, and hair is a big part of who we are. In terms of different causes of hair loss, I like to break it into 4 different categories. It's hormonal, nutrient deficiencies, environmental, autoimmunity, and a lot of it's inflammatory related. In terms of current treatments that we use for hair loss and what I use in my practice, they include supplements, drugs such as finasteride or dutasteride, oxides such as minoxidil or Rogaine, as is commonly known in the States. I also use a lot of PRP, and some of you, I'm sure, use stem cells and exosomes, and I'm sure some of you in the crowd also do hair transplantation. So in terms of hair loss, nutrient deficiencies, iron, biotin, B12, we know that there's a lot of growth factors that affect hair, right? And for many, many years, PRP has been a great source of growth factors, right? At least that's what people think, right? People think, oh, you take something from your blood, you spin it down, and you have all these goodies in there. Granted, you have certain growth factors such as PDGF, TGF-beta, FGF, VEGF, EGF, and so on, but those levels are very, very, very low, and I don't think many people realize that. In addition, you have a lot of variability from person to person. So one male to one female, and one session to the next session, your platelets are going to react differently. All these levels will be affected differently. But with that said, PRP has worked really well, and I'm sure many of you use it in your practice, both on the skin and on the hair. So there's no denying that. But so let's move on to environmental factors, oxidant stress and inflammation. That's a big part of, I think, kind of an underappreciated, just an area where it's not really looked at too much, right? We know that oxidant stress, both on our face and our hair, can adversely affect hair follicles, right? The role of reactive oxygen species in hair loss leads to increased free radicals, which is associated with DNA damage, damage to RNA proteins, basically resulting in premature aging and cell death. So reactive oxygen species has a central role, and I've incorporated antioxidants into my treatments to help with that, and I'll show you some data. But what the antioxidants really do, and this is what I postulated, is it increases nitric oxide availability, right? In particular, antioxidants helps maintain NO bioavailability, decreasing oxidative stress within physiological and pathophysiological processes, ultimately leading to more robust hair. This here is just some primary data. I think the most important data, most relevant, is the graphs on the left. It's cell proliferation data of dermal papillae cells, where we treated them with different conditions. We had just standard growth media, media plus growth factors, namely EGF and FGF. And then thirdly, media plus growth factors was EGF and FGF, as well as antioxidants, which include glutathione and L-arginine. We're able to see at normoxia, you kind of see a nice dose response in terms of increase in cell proliferation. But when you subject these DP cells to hypoxia, which is at the bottom left, you see a markedly increase or a markedly difference in the cells that are treated both with the growth factors as well as the antioxidants. Moving on to other areas such as inflammation, right? So what I really want to transition and change gears to is, PRP has worked really well. With exosomes coming onto the scene, people are using it for everything. People are using it for skin, people are using it for hair, et cetera, et cetera. But I really want to know, what are the fundamental differences that are causing these results that we're seeing? In terms of hair growth, how exosomes work, right? We know they have certain growth factors, IGF, KGF, HGF, et cetera, et cetera. And they have several microRNA that play important roles in hair growth. Different sources of exosomes, but in particular, what we're going to talk about today are the adipose-derived mesenchymal stem cells. So this is from my lab, which is a third-party, independently run, aside from ExoCoBio. I just wanted to see the results for myself. Looking at these three growth factors, VEGF, HGF, and FGF2, this is just relative absorbance. In blue are adipose-derived exosomes from adipose-derived mesenchymal stem cells, and in orange are platelets, right? You saw that third or fourth slide where I showed the growth factors that are involved in that exosomes from PRP, right? There was PDGF, VEGF, FGF, but you see here when we're looking at, we're trying to quantitate levels of that, right? We can't even pick anything up in terms of relative absorbance when we're using or we're running our ELISAs. So we can see here in terms of these three growth factors in particular, VEGF, HGF, and FGF2, you have higher levels of growth factors compared to exosomes that are derived from platelets. Now, we talked a lot about in previous talks about the inflammatory response and the anti-inflammatory properties of exosomes, right? So here, what I wanted to do is I wanted to look at a whole slew of inflammatory markers and looking again at exosomes from adipose-derived mesenchymal stem cells in blue, and then looking at exosomes from platelets. I think with the exception of IL-6, you see higher levels of inflammatory markers, 2, 5, 15, TNF-alpha, TNF-beta, in exosomes derived from platelets as compared to exosomes derived from adipose-derived mesenchymal stem cells. So in a nutshell, you could say, and you can conclude that exosomes from adipose-derived mesenchymal stem cells, they have a lot more growth factors and they have intrinsic anti-inflammatory properties. So here we come to this, right? We come to the Benev exosome regenerative complex, right? And I started incorporating this into my procedures in addition to a lot of other modalities that I already talked about. And this really helped boost and push my results further, right? And the title of my talk was just alopecia. It was a generalization, right? But I treat patients with alopecia areata, alopecia totalis, androgenetic alopecia, and I use the same kind of formula and combination, and adding exosomes have really pushed my patient satisfaction level and results to another level. So these exosomes in particular, they're stem cell derived, they have potent growth factors, peptides, enzymes, amino acids. In addition to their intrinsic anti-inflammatory properties, there are antioxidants added along in the exosome regenerative complex. So I'm just going to go over 3 quick cases. This is a patient with alopecia areata, and again, using just a multimodal approach to hair loss. This was a 32-year-old patient who went to a dermatologist, went through the intrafollicular steroid injections for many, many months, no results. It actually got worse before it got better. I incorporated all of these modalities together, and in about 5 months, seen pretty much total reversal of this condition. Another case is alopecia totalis. So this was a young female in her early 40s, had very, very beautiful hair, and just came to me and looked like she just lost everything, right? So again, initiating all these different multimodal approaches in addition and including exosomes, basically over the course of 9 months, be able to totally reverse her condition. This here is an African-American female with androgenetic alopecia, basically here on the crown, and basically, again, using the same approach, in incorporation of exosomes in the course of a few months, being able to really improve her outcome. Same patient in terms of the kind of the frontal part of her hair, which is very difficult to treat in patients in general. Again, and this is just microneedling the exosome regenerative complex. So closing remarks, hair loss is multifactorial, right? Exosomes are great, right? We know that exosomes have a lot of growth factors, have a lot of anti-inflammatory properties, but you have to approach it from many, many different kind of prongs, right, in terms of treatment, right? We know antioxidants increase nitric oxide levels, which increase oxidative stress on the hair. Combination of growth factors and preservation of NO bioavailability leads to better hair growth in patients. But I think the icing on the cake for me is that exosomes provide the necessary growth factors, and their intrinsic anti-inflammatory properties have really, really pushed my results to a whole nother level and my patient satisfaction to a whole nother level. Thank you very much.
Oh, okay. Okay, thank you, Jim, for your talk. And now we're going to have about Dr. Todd Schlesinger from the US. He's going to talk about extracellular vesicles in aesthetics. Is it fact or fiction?
Fact or fiction, thank you very much. And thank you for hosting us and for everybody for being here. So I'm Dr. Schlesinger, Todd Schlesinger. I'm a dermatologist in USA. And these are some of the devices I have in my practice. I'm going to take you through a little bit of a rapid fire through some of the science of exosomes, sort of facts or fiction. We'll talk about, exosomes today among extracellular vesicles. You can see the size. These are the ones we'll talk about today. You can see the size of these and how they get taken up into cells. So the green are exosomes that are very near a nucleus and the cytoplasm of a cell. So they are taken up an exosome into cells. You can see what size they are in the upper left-hand corner, focusing around 100 nanometers. So they diffuse, exosomes diffuse through tissues. They find their target, they have a function, and they're able to affect changes in the skin. When you think about stem cells and exosomes, you think about very differently that exosomes go through tissue, they can be stored, they can be transported easily, as ExoCoBio has figured out how to do for us, versus stem cells, which are living cells, and they're a little bit harder to use and have limited therapeutic potential. Tons of research going on. You can see it's exponentially increasing around the world. There's hundreds of thousands of mRNAs, hundreds of thousands of lipids, and we focused on what's in dermatology. So how do exosomes work in dermatology? These are some of the growth factors and things that we think about in our field, and we think about the science of exosomes and how they're going to affect us. We're looking for helping blood vessels. We're looking for scar improvement. So cells uptake exosomes, as you saw. These are some of the ways in which some of the science behind how that's done. Multiple different mechanisms of action are there. And when you think about making medications and drugs, especially neurological drugs, they don't perform as well in the clinical as they do in the lab. And reason why is they may not get to their target. So exosomes can help medications get where they need to be in our body, as opposed to crossing the blood-brain barrier. We already know many of the sources that exosomes come from, and this is exosome characterization, so how you actually determine what is in an exosome, what's there, or how you determine if your exosomes are actually what size they are. So these are all the different techniques that you can use. So these are some of the exosomes that are available in the United States. You can see the separation technique and the concentration here of the different products. Adipose seems to be one of the biggest ones that we're using, but human-derived is not always available around the world. How they're made and how they're isolated. This is important. Looking at these companies and looking at how things are done, ultracentrifugation has always been the gold standard, but tangential flow filtration has really risen to the top, and then immune affinity might be the next step. It's not quite ready for the prime time yet, but it's the next step. And this is a new technique called double tangential flow filtration, using a membrane with a serpentine interior to help try to increase the efficiency and purity of production of exosomes. This is called immunoaffinity, another immunoaffinity. This is another way of using CD63. This is a heat process. So you capture exosomes, you heat and release. So this can help further improve the viability and actually the purity of the exosome you derive. So we already talked about the industry. So these are some of the claims. So these are all the things we think about in dermatology, in beyond dermatology, in medicine in a general sense. But how much of it is actually true? So I'll go through some of these things pretty quickly. So in dermatology, we're really interested in all the things we can do, especially when it comes to orphan diseases. We have a lot of severe diseases. If you've already seen some excellent outcomes from those. And this is atopic dermatitis. So look at some of the science. This is all just background science. So inflammatory cytokines reduced. Reduce epidermal thickening. So how can that be done? Increase hydration. That's something we want to do for atopic dermatitis as well. And we also want to increase ceramide synthesis or protein synthesis in the body, in the skin. So here's a product. I'm just going to go through different products. This is not, it's just to show different things. This is a product that shows it can do this. It can hydrate and fortify skin, and that would be through these two mechanisms of action. Anti-inflammatory, this is also something we really want to look at. This is the mouse model, it's a psoriasis model. So they've taken imiquimod, they've created a mouse model increasing IL-17, which as we know is a problem for psoriasis, one of the cytokines, and they use exosomes to try to reduce that in this model. So looking at the science, we may be able to use psoriasis, exosomes for psoriasis in the future. What about stretch marks? This is another big, scars, healing scars. Here's a mouse model. This is a control. These are non-diabetic exosomes you can see produce a healing effect, and diabetic exosomes which do not. And if you actually knock out a gene in these cells that produces apoptosis, if you knock out that one, you get improved wound healing. So, modulating tissue and improving that can actually help you in even diabetic patients. Here's a study for scar repair and prevention. So you can see reduction in scar volume, pore volume, and roughness. This is for carbon dioxide, for acne scars. This is injection of exosomes, and we don't inject exosomes, but this is just in a mouse model with a burn scar showing that subcutaneous exosomes may help in the future. Now there's no injection of exosomes now approved, but in the future that may be something we could do when they actually become a drug in the future. So angiogenesis, here's another thing. We want to make our flaps do better. We want flaps to improve. Here's a model using human umbilical vascular endothelial cells stimulated, stressed by hydrogen peroxide. So this is how you get exosomes out of these. And what does that do? That can help our flaps do better by improving survival, by promoting angiogenesis. Hair loss, you've already heard a lot about hair loss, same effects as stem cells, improving hair counts. And you can see before and after there. All this different published data from around. Here's another product. What does this one claim to do? This one claims to reduce pigmentation. So here's keratinocyte-derived exosomes that can decrease melanin and decrease tyrosine production. So this is just a slide showing you a marker, what they look like when they're taken up by the cells in our skin. Anti-aging, this is the biggest one that we think about. Of course, you've seen this slide before, something that ExoCoBio and Benev have been very focused on, and others, and this is that study with using fractional carbon dioxide laser. So improving the outcome of our procedures is what we want to do. This one shows a percentage of scar reduction in a CO2 laser with and without using exosomes. In skin patients, dupilumab, redness, it's one of the side effects we see from biologics. Facial redness improved with exosomes as well. So just some of the different things that are out there. Here's a before and after. This is PRP on one side and then exosome on the other side showing the difference in erythema. So that's also helpful. It plays well into our other inflammatory skin diseases that we see. Of course, the regulatory landscape, as you know, the holy grail, everyone wants to be in the green, but in reality, we probably are in the red, getting to the point where these will eventually become actual drugs that can be actually used under approval by FDAs around the world. So these are some of the challenges in exosomes. How do we make them scalable? How do we make them work well? And how do we make them pure? And how do we get them out to everyone, regulatory-wise? This is one of the things, safety. So where the exosomes go, how are they cleared in our body? Our body clears them out through different things. The lungs, the liver, and the kidneys are one of the three major places where they're cleared. So we have to be careful about applying exosomes to make sure that they're safe, especially if they're going to be used more in a drug-like manner in the future. So how do you do that? This is just some other science. You can mix them with polymers to slow down absorption. You can incorporate them after gelation in a polymer, or you could do both. You can do both together. So these are just different things that can be done that when it's in the future, maybe it's 5, maybe it's 10 years, when exosomes become more of like a drug, more used for targeted delivery for skin disease or other diseases, that we can actually see that work in a more healthy manner. So just a few cases from my practice. Acne scarring, we're getting great results using Sulfamex in combination with their exosomes as well, so that's kind of nice to see. Just some skin smoothing. This is showing that you've got some skin quality improvement here. And then this is one of my prize cases. I really like this one. This is just Sulfamex, just exosomes, no fillers, no modulators, nothing else. Just shows you the pure outcome of this tightening that we're getting. You can see in the lower face, 2 treatments. We see the improvement. We know it's working for our patients. You can see skin quality improvement, and you can see that jawline improving. That's really the holy grail that a lot of patients really want to see. That compared to a carbon dioxide laser case, again, using exosomes after. This is just straight carbon dioxide, looked at it about 8 to 12 weeks later. And you can see, actually, I wouldn't say she's got improvement in the lower face, but I like the tightening from the Sulfamex and exosomes combination better. I like the skin quality that she got from this. So it's comparable, it's helpful, it's been a great thing for our practice. So thank you very much and appreciate the time.
Thank you very much, Dr. Todd. And now we're going to have Dr. Suparu from Thailand, which I just know here in our dinner. And he's going to talk about a multiverse of applications in dermatology. Welcome, Dr. Suparu.
Good evening, everyone. It's really my honor to be here. The dermatologist from Bangkok, Thailand. I will try to focus on the clinical application and clinical study of the multiverse application of the exosome. First, about acne scar, you can see that this is on my forearm. I try to use the exosome and also the control gel. We can see that exosome help reduction the downtime very fast. We can observe that since the 3 hours. So how that's supported by the first randomized clinical trial of the exosome and fractional carbon dioxide laser in the treatment of acne scar. That show that better efficacy and shorten post-treatment downtimes after fractional carbon dioxide laser. And how about the other treatment of acne scar? Fractional picosecond laser, fractional microneedle RF. So this case, day 1 and day 7 after fractional picosecond laser with the high fluent. This is the control with moisturizer, with ceramide, and the others was treated with exosome. You can see the significant improvement difference. So from day 1, the patient can see the significant difference. This is without exosome, and this session with exosome. You can see the patient downtime short from 7 into 1 or 2 days. This is day 1, day 3, day 1, day 3 with exosome. You can see the shorter the downtime. Also, I perform the split lesion. This side, I use the fractional picosecond laser first, and you apply the control moisturizer. And this side, I use the exosome. You also can see the shorter downtimes. And how about the efficacy? This is just 1 or 2 cases of mine, so long-term study are required, but the patient feel that significantly decrease the downtime and also show the better efficacy, but we need larger study. And how about we not only for the skin booster, we have the polynucleotide intradermal injection. So split face to see that this receive picosecond laser with exosome topical application. The other side was treated with picosecond laser and polynucleotide intradermal injection. The downtimes almost just not significantly different, but the patient they feel that he had previously treated with the picosecond laser before, but this time he feel that the downtime are significantly reducing. However, for the topical exosome application, he feel no pain from the injection like the polynucleotide size. This is the day 1, day 3 on the topical exosome, and the other side with the polynucleotide intradermal injection. I think the downtime almost the same. And the others treatment with the fractional microneedle RF, I use quite high frequency. For nasal scar treatment, I perform the same. This time, a topical exosome application, and the other sides will fractional microneedle RF, and then topical intradermal polynucleotide injection. This saw that the shorter downtimes also. Fractional microneedle RF less have the downtime than fractional picosecond laser, but this also shorter downtimes. So you can see that topical exosome, shorter downtimes like the polynucleotide injection, and also no pain from the injection like the polynucleotide. And when the patient, and I also absorb that the more of the exosome application times, the more skin quality improvement. You can see this is no exosome. This one, first session, second and third session. So this is the same power. So the patient downtime significantly decreasing, and also the patient's skin quality shows significant improvement. This is the daytime post-picosecond laser. On the third session of exosome in combination with post-picosecond laser, skin almost improved like the no downtime at all. So how about the other type of scar? Hypertrophic scar and keloid. I added the exosome into the standard treatment, topical picosecond laser, fractional laser, topical steroid, and add the exosome. They are show more benefit add on to the traditional treatment, especially the itching. the patient feel that significantly itching. This is the hypertrophic burn scar. Previously treated with standard treatment with the laser. When add one treatment with exosome, patient feel significantly decreasing in the itching and also the redness and size of the scar much reducing. And this is the 2 sessions of the laser and in combination with topical exosome and topical steroid with no injection. You can see the improvement. So that could explain that by the molecular study, we can show exosome have the effect on first stage on the wound healing and also the scar formation. So I think the early application of exosome could help the patient a lot. How about the others? Atrophic scar. This is the POC scar. This is only 3 sessions with the substitution, laser, and also exosome. The patient feel very impressed with the result. And this is a surgical scar revision. So the laser and in combination with the exosome and substitution, almost they say that when they apply only minor makeup, they cannot see. The stretch mark also, only 1 session with laser and exosome. This is 2 sessions. So from my practice, the thing that I found interesting is about exosome and hair loss. It's quite impressive. In the case with currently on the treatment with oral medication, fractional laser or PRP, when I got the exosome and apply first, second, and third session, the patient feel improve in the hair thickness and hair density, even the patient with previously medical treatment. So the other case with the no previous treatment, I combine with the micro-needling and topical exosome. So this is only 2 sessions. And the interesting thing, as all the speaker mentioned, is about the effect on the gray hair. The patient feel that they have to go to have the hair coloring. If every one month, something like that, this is 2 months already. So I think there need more study. The other techniques that I use, the fractional picosecond laser in combination with the exosome. The benefit of the fractional picosecond laser, there is almost no pain, no downtime, like fractional CO2 or micro-needling. This frontal area also show the improvement. This is for 3 sessions, no other medication. So the other thing is, even for the case with previous hair transplantation and autologous hair tissue micrografting and on oral medication, fractional laser, PRP, or low-level therapy, when you add exosome treatment as the topical after you use the laser, they still have the improvement. So I think it's quite interesting. They're supported by 2 clinical trials. This is use the micro-needling with exosome for 12 consecutive weeks. So increase the thickness and density. This is I'm looking for the result, but this is just the abstract. So that micro-needling with ASCE exosome show the improvement. How about the pigmentation disorder? So I have to say that it's quite difficult for me. We need many treatments, sun protection, something like that, laser, but some cases show the improvement. But for me, as we in Thailand, the strong sun, something like that, so the result maybe equivalent. But there are studies that show that in the treatment of melasma, the stem cell-derived exosome in combination with non-ablative fractional laser, micro-needling, or plasma device can also achieve the good result. And the other study also combination with the human adipocyte-derived stem cell exosome in combination with the micro-needling can help improving the facial skin aging. This is a study. I feel that exosome is not for the aesthetic only. It's for the skin disease also. This case, severe recalcitrant seborrheic dermatitis. He previously needs some of short course of oral steroid, pimacrolimus or topical steroid, recurrent. And at this time, I use the ASE exosome with the electroporation, not needle. The symptom of itching and redness of the patient significantly decreased during the first 2 or 3 days. And later, I give him the maintenance treatment with the ExoBalm. And this is the 3-month follow-up. He don't have any symptom and skin quality. significantly improved. The other difficult to treat skin diseases is the lichen amyloidosis. Refractory case, they previously on high potency steroid, tretinoin peeling, something like that. When I add topical exosome application for 7 days, the symptoms significantly improved and the itching decreased from the score 9 to 10 to 2 to 3 itching symptoms. So this is the role. So I will try to summarize that this is the study that support my information. Dexamethasone, they improve the efficacy and shorten the post-treatment downtime. Fractional CO2, maybe PICO, fractional mechanical thermomechanical ablation, and improve the downtime. There is maybe the topical application improve the downtime comparable to polynucleotide intranormal injection. For the hypertrophic scar, atrophic, and stria, they improve. They can use in combination with the laser like this, or and also standard medications such as topical triamcinolone acetonide 5-FU, or used as monotherapy. The other interesting is about the AGA. They have the efficacy. Even in the previous case with hair transplant, autologous hair tissue micrografting, PRP, fractional laser, they may, I think they have the efficacy on the gray hair reversion. Can use as the mono or combination with the laser and treatment. There are many techniques that are published in the journal. Also benefit on the skin aging, melasma, and for the recalcitrant, difficult to treat seborrheic dermatitis. And as for my interest, I think lichen amyloidosis may have the role. So, final slide, I would like to conclude that multiple efficacy of the exosome application, both in aesthetic and medical dermatology. However, we need more study. As I reviewed, this is only 4 clinical study of the exosome currently. We need more basic and also clinical study to verify the efficacy and also the safety of the exosome for the clinical application. Thank you. And 감사합니다.
Thank you very much, Dr. Suparu, for your experience. And now we're going to call our last speaker in this session, Dr. Lidia Madziuska. She's from Poland, and she's going to show her experience about treating scar treatments, skin atrophy treatments. After the session, we're not going to have questions, right, Diego? Okay, so we go after, you keep your questions for the next sessions. We will do it all together in the end of the section 4. Okay.
Good evening, everyone. It's a great pleasure and a great honor to be here. Before I will talk about the case that was my case that was awarded, I would like to say a few words about plant-derived exosomes or extracellular vesicles as the product I use in my practice. This is ASC Plus, ExoCoBio's product, which is based on RAES stem cells exosomes. So as you probably noticed in the recent years, the number of publications on plant-derived extracellular vesicles is constantly growing, but it's still hundreds compared to thousands that we have on animal-derived exosomes. So when you go to the clinical trials, registered clinical trials, you will see that there are not so many registered trials, the ones that would explore plant-derived exosomes. Researchers usually work using ginger, grape. These are products that have been known in traditional medicines for ages for their anti-inflammatory, antioxidant, and antibacterial properties. But one of the main challenges in this kind of trials is a good quality product, GMP grade product. So this is a challenge that we are facing. This is an example of such research. The researchers here plan to use grape seed exosomes in curing or treating oral mucositis, which is associated with chemoradiation treatment of head and neck cancer. Another interesting publication, this is on animal model though, but I'm showing this because, results are quite impressive. And this is the trial where the researchers used ginger-derived exosomes or extracellular vesicles, and they were able to abolish severe pneumonia, which developed in the course of SARS-CoV-2 infection. So the result was really very satisfying. Well, actually, ginger has been used in the number of studies, and again on animal models. Its efficacy was proven in, for example, chronic or acute colitis or IBD. So the results here are promising too. And then RAES. RAES, again, in traditional medicine has been known for ages for its anti-inflammatory effect. It's been widely used in cosmetics, mainly due to its skin brightening and skin tightening effect. And it's been also used in numerous indications, as you can see here. It contains, in cosmetics, was important, it contains flavonoids, phenolic acids, and other phenolic compounds. So its efficacy in cosmetics has been known for a long time. This is an example of a publication where it was shown that rose petal extract had anti-inflammatory effect on the skin that was exposed to UV radiation via, this effect was achieved via reduction of MAPK signaling pathway. So also very promising results. The main challenge that was also mentioned before by other speakers regarding exosomes is quality of the products, its purity and certain standardization. And as it comes to the plant-derived exosomes, this is even more difficult compared to the animal ones. So this is data that we have from ExoCoBio, manufacturer of AC+, and it shows rose stem cells exosomes, so nano-tracking analysis showing size of the size distribution of exosomes, and also confirming that the membranous vesicles, and as you can see, the size and shape resembles that we know to ASC exosomes. And again, cellular uptake of rose exosomes in human dermal fibroblasts, we can see that they would promote migration and proliferation of the fibroblasts, thus increasing production of the collagen. Melanin content, well, we know rose for the skin brightening effect, and it was shown here that the rose stem cells exosomes could reduce melanin content in the cell. But this is the most exciting thing about exosomes, isn't it? microRNAs. I think that still a lot of things to be researched, and it's a peak of iceberg, as Dr. Gold said, but as you can see here, regarding our the product that I use, 27 types of microRNAs match with human microRNAs. 6 of them are responsible for migration and proliferation of the cells. So with plant microRNAs, it's difficult because little more than 400 were described, but still we know that pre-microRNA sequences have not been, are not available or incomplete for most plant species. So a lot of research have to be done to identify them. And the product that you perfectly well know, this is lyophilized stem cell extract with other active compounds and activating solution. Where do I use it? Well, we all agree, all speakers who presented their cases, that this product, application of this product would speed up the recovery process. This is a neck, 37-year-old male patient, one side, and fractional ablative razor, one side treated with ASC plus, the other one left untreated. The results after 6 days, you can see the difference between the sides. And going to the case that I was going to show, scar treatment and skin atrophy treatment, well, we all remember the healing process, wound healing process. So please turn your attention to this inflammation stage with the release of the cytokines. And then we have proliferation and remodeling and scar formation. So remember, inflammation and remodeling, this is where exosomes would act. And all the publications that I was able to get regarding scar formation and wound healing were based on exosomes from adipose tissue. That's why I'm showing this mechanism which is behind it. So we still have to work and do some more research how the plant ones would exactly work. But what is important here, exosomes would act in the wound stage and in a scarring stage. And in the wound stage, they would upregulate the expression of the genes collagen responsible for the production of collagen 1, collagen 3, CD34, decorins, elastin, keratinocytes. And parallelly, as you can see, that lower levels of alpha smooth muscle actin and TGF-beta1 would prevent or would optimize the level, the differentiation of fibroblasts into myofibroblasts. Whereas in the scarring stage, we have downregulation of the genes responsible for collagen 1, collagen 3, beta smooth muscle actin, protein P38, TIMP. The same alpha smooth muscle actin and TGF-beta 1 are low and prevent differentiation of fibroblasts into myofibroblasts, which prevents from overgrowing of the scar. So to make it short, exosomes would promote wound healing and reduce scarring. And this is the case that I wanted to show you, the awarded ones. This is a patient, 36-year-old female patient. She underwent skin graft. She had a skin graft when she was 6, and it adapted very well, but she was looking for ways to improve the way it looked. It's the middle part of her face. She didn't want to conceal it with makeup all the time, so she was desperately looking for a solution. She had 6 sessions of collagen injections. I don't know what the product was. And apparently it stimulated the site that the scar overgrew. It became hypertrophic. The skin of the graft became thick, and it was clearly visible. So treating the physician who treated her injected steroids, and which was followed by classical symptoms of steroid injections, skin thinning, skin atrophy, and discoloration, increased visibility of the capillary vessels. When she came to us, we decided to do microneedling and application of ASC+. She received product to apply it at home at least twice daily. And she came back after 7 days for another microneedling session and application of the product. And you can compare the effect after 12 days of using a product. We noticed even skin tone, there was a reduction of the margins of the scar, reduction of visibility of those small blood vessels. Color improved, while the skin was smoother, seemed more hydration. There was not that pulling effect that this is what patient reported. So since she was a very demanding patient and very unhappy and difficult from psychological point of view. So the fact that at the end she was satisfied, I was satisfied too. So this was the last slide of my presentation. I would like to thank ExoCoBio team for inviting me here, for having me here. I would also like to thank our Polish distributor for our fruitful, I hope, cooperation. Thanks a lot, and I was going to say have a great evening, but a lot of presentation is ahead of us. Thank you.
Okay, thank you for all speakers. And now I'm going to invite Dr. Tingling to be here to help us with section 4.
All right, so as Dr. Lim Ding Song gets into the podium, we're going to have the next case, Dr. Suramis Estrada Blanco. She's actually from Spain, but she couldn't be here tonight, unfortunately. So we're going to play the video. This is a very special case because it's one of the first ones with our new product, RLB, for the female intimate rejuvenation. So let's take a look, and then we will start with the speakers here in the room.
Hello, everybody. First, I would like to thank ExoCoBio for the possibilities of sharing my clinical case in this amazing event. Now I'm going to try to share my screen. Yes. Well, my clinical case is about exosomes, of course. Exosomes, new solution in female genitalia regeneration. Lichen sclerosus, formerly called lichen sclerosus atrophic, is a chronic inflammatory disease of the skin characterized by the presence of papule or atrophic, white plaques of known etiology, which primarily affect postmenopausal women. Prevalence in general population is estimated at 1 of every 300 to 1,000 individuals. There may be a trigeminal lesion, but it more frequently or most appear in the genital area. Our clinical case was 60 years old patient with history of hypothyroidism receiving oral treatment with uterus 50 milligrams daily, one vaginal delivery 35 years ago, and menopause was at 50 years old, started with symptoms of itching, genital burning, and difficulty in intercourse due to dyspareunia 5 years ago. After previous diagnosis of sclerotrophic lichen by dermatologists and treatment with topical corticosteroids and various moisturizers without improvement, come to my consultation for another opinion and looking for another solution. After all these symptoms and good physical examination in the vulvar and vaginal area, I diagnosed this case as severe lichen sclerotrophic. The protocol was local anesthetic cream to the entire vulva for 40 minutes before the treatment, 2 sessions every 4 weeks of local fractional bipolar radiofrequency energy therapy with gold microneedles. After each session, I administer exosome IC plus RBL to entire damaged vulvar area, to entire it in labia minora, labia majora, clitoral hot, vestibular space, perineal space too. After starting treatment session, it was recommended to avoid chemical products such as sauce or gel in the vulvar area. After the first session, she began to work vulvovaginal epithelium with vaginal dilators and has managed to sexual intercourse with her partner. The patient currently reports improving her itching and stinging symptoms, and the most important for me was no adverse effects or complications were recorded at the end of the treatment in this clinical case. This is the picture before the treatment. Looks here in the vulvar area, the white plaques. Look here, the abrasion here in the near to urethral space. Look the signs here in the vestibular, the sign of the vestibular craurosis. This is the picture after the treatment. Look the improved or the lesion in the vulvar area. And this is the same picture before and after treatment. The conclusion in this clinical case was the inflammatory dermatosis of the vulva constitutes a frequent reason for consultation in our medical practice. And it's very important to the degree of injury and correct diagnosis before the therapies. The exosome AC plus IBR applied after local analgesics in the tissue seems to be a good regenerative therapy in this pathology. Clinical follow-up of this patient should be carried out in the long term in order to achieve remission of the lesion, control the disease, and avoid future complications. Thank you very much. Thank you for the opportunity.
Well, thank you, Dr. Zoramis, for such insightful sort of sharing. So we'll continue on with Dr. Ajang Yasdani from Canada to share with us the decreasing laser recovery using exosomes, please.
Oh, I see. Okay. Well, thank you very much. And I'd like to thank ExoCoBio, Benev, as well as our Canadian partners, Excite Tech, for inviting me to come speak today. My name's Arjang Yazdani. I'm a plastic surgeon from Canada. Sorry. Oh, okay. Is this better? Okay. My name's Arjang Yazdani. I'm a plastic surgeon from Canada. I'm here today talking about the use of exosomes in ablative laser recovery and how to improve our laser outcomes. We've been using exosomes in our laser recovery, particularly with our ablative patients, to really improve the outcomes, to quicken healing, and also to decrease the risk and the incidence of redness post-ablation. I've been doing lasers for about 20 years, and I've used CO2 as well as erbium. And I can say that while we've gotten better at diminishing the risk of redness, swelling, pigmentation, post-ablative resurfacing, it's still a significant problem, and it affects how our patients get their outcomes, the quality of their outcomes. The patients who do have this problem end up having multiple treatments later and are dissatisfied with their results. So we've, even despite our best protocols, this is still an ongoing problem for our deep resurfacing patients to some degree. We've incorporated the use of exosomes in these patients and have been quite satisfied with how it's improved the risk of these problems. This is an example of a patient who's got this problem. So she's had a deep erbium resurfacing of her perioral area. You can see in the first picture, she's back at day 1 for her debridement and her nursing care. At 1 week, she's cleaned up, she's almost healed. It really does look good for such a deep resurfacing. But at the 3-month mark, she comes back and she's got redness, she's got some pigmentation. This is a time when that result should be excellent. The texture and the wrinkles are better, but you really can't appreciate it because of that redness. So it really diminishes the result, makes the patient dissatisfied. It makes us have to continue care and add other treatments to try to improve this. So we've incorporated exosome treatment in our post-laser recovery. And as you all know, it's a simple thing to do. Right in the procedure room, we'll put on the exosomes and give the patient half the bottle to go home to apply later that evening. And we find that that has improved. There's a video here, but just for the sake of time, I'm just gonna skip past it. We've had such good outcomes. We've actually used our outcomes as a basis for a clinical trial that we're running. This is a randomized controlled, double-blinded, placebo-controlled trial that's ethic approved. And we've actually begun this study. It's powered for 54 patients with 3 arms, a full face arm, a split face, as well as a control. I'm not going to be talking about the results. We're not complete with this study, and hopefully next year at this symposium, I can present the results of this study to you. But today I'm just going to go through some of our patients that have had exosome treatment post-deep ablative resurfacing and really show you how it's improved their outcomes. So this is our first lady. This is a lady who's had a full field erbium resurfacing, a deep resurfacing at 800 microns. She's back. The first picture is her 1 day return back to clinic for her debridement, her nursing care. The second is her 1 week visit. She's healed, but she's still a little bit red. At the 2 week mark, that redness is gone, and she looks great at the 6 week mark. So we tried to really limit that healing to the 1 to 2 week mark for our deep resurfacings. We think that's the limit of healing that the patient can tolerate. And having these exosomes on these patients ensures that they really do have that healing complete within that 2 week mark. This is a lady who had profractional laser resurfacing. Again, a fairly aggressive deep resurfacing at 22% with 2 passes. Her 1 day visit is back. At 1 week, she's still red. At 2 weeks, that redness is completely gone, and she looks great at 6 weeks. And again, this is a patient who's got the profractional treatment on the procedural table. She gets her exosomes and gets half the bottle to go home with afterwards. This patient, some of you may recognize, is a beautiful young lady who came to see us 6 weeks before her wedding and told us she wanted her eyelids resurfaced. And normally I'd say no, but she managed to twist my arm into this. So we really felt the pressure. We wanted to do a deep resurfacing, but we had this pressure of a wedding coming up, and so we applied the exosomes. Gave her the exosomes to go home with, and really this is her 7-day journey. It's a deep profrac resurfacing of her upper and lower eyelids, 2 passes at 22%, and at 7-8 days, you can see that she's completely healed. A little minor redness, but really for the aggressive nature of that ablation, it's not bad at all. This is a young guy who had a full field resurfacing of his lower eyelids, 2 passes, deep resurfacing, exosomes were placed immediately afterwards, and this is his 1 week result. And this is really a great result. This is probably one of our best, and something I can say that we wouldn't have seen prior to having exosome treatment on these ablative patients. This patient had profrac treatment, again, an aggressive profrac treatment, full face, 2 passes at 22%, followed with full field perioral resurfacing at 400 microns. Exosomes were placed immediately afterwards, and she was given her vial to take home. 1 week, you can see that she's still a little bit red. It's not too bad given how aggressive she was. At the 2 week mark, she's healed, and at 6 weeks, she really looks great. And finally, this patient had problems with redness. We knew she was at risk of having further redness post-ablation. She was a full face profrac, aggressive at 22%, 2 passes, followed with perioral full field at 400 microns. Exosome treatment immediately afterwards, as well as going home, and this is her result at 2 weeks. So no redness, still a little bit swollen. She's got some maturation to do with that resurfacing, but she really does look great. So in conclusion, we've really incorporated the use of exosomes in all our ablative resurfacing. We also, in fact, have been so satisfied that we use it for our aggressive non-ablative resurfacing as well. We find the exosomes can help laser recovery, decreasing redness, and in some cases also decreasing that potential hyperpigmentation. It gives the patient better results. It gives us more confidence to give aggressive settings, and that's the big thing. If we know that we can shave off a couple days of their healing time, as well as decrease that risk of redness, it lets us be more aggressive with our settings, and we get better results, and patients are happier. And finally, I think there is a role to expand the use to other energy devices outside of the ablative lasers, and we're starting to do that. So thank you very much.
Thank you very much, Dr. Ajang. And because of time, we'll move on to the next speaker, which is Dr. Jennifer Levine from the United States, which she will talk about using exosome for skin. Please.
Yes. All right, so I'm going to be talking about something a little different, which is using exosomes in treating vascular occlusion. I know it's a little late, but nothing gets the blood pumping like a vascular occlusion. I am a facial plastic surgeon in New York, on the Upper East Side, and I'm so excited to be here in Korea. And thank you to ExoCoBio and Benev and Ethan Bin for having me here. Here are my disclosures. We'll skip this. So here's my lovely patient. She actually lives in Denver, Colorado, and she has an apartment in New York. And she calls me up on a Monday. She calls my office up and she said, you know, I had some filler injected when I was in Denver about 5 days ago, and could you help me out? I have an infection. And I said to my office staff was like, you want to call in an antibiotic for this patient? I said, no, I want to see this patient. So she didn't want to come in. We then convinced her to send a picture, in which case I had her come in immediately. So she had calcium hydroxyapatite injected into her nasolabial fold approximately 5 days prior to this photograph. And if you weren't sure what the vascular distribution of the angular artery is, you can see it quite clearly here. So she was, you know, and what did I do is I did whatever I could. So I treated her with Hylenex, normal saline. I did see some reperfusion. I treated her with antibiotics. I gave her a Medrol dose pack. I gave her Cialis. And I decided that because there were early signs of impending necrosis or even some necrotic tissue, that I was going to treat her with exosomes. And because we did not have the ExoBomb yet, I took the rest of the exosomes and I mixed it with Aquaphor, and I instructed her to apply that to the area twice a day. And this is her 48 hours after treatment. She lived in University Place, which is really close to my office on the Upper East Side, and I really couldn't convince her to come in because she said, I'm doing great, you don't need to see me, I'm fine. And here she is a few months later when I did see her again. So that's my case.
Fantastic. It was interesting. All right. Thank you very much. So next we'll open the Q&A later. So next speaker will be Dr. Carlos Garcia Miras from Mexico, and we'll talk about hair.
Okay, good evening, everyone. Buenas noches a todos in Mexican Spanish. I'm excited to be here tonight, sharing my experience with exosomes developed by ExoCoBio, Benev, with the treatment for androgenetic alopecia. Let's dive into the case of a 37-year-old patient with androgenetic alopecia grade 5 of Hamilton-Norwood. He had been struggling with hair thinning in the interparietal region for 5 years, affecting his appearance and well-being. Seeking help, he came to our clinic, and he doesn't have previous treatments and a family history of alopecia. In the physical examination, revealed frontoparietal region, miniaturized in both apex, vertex, and interparietal region, and addition size of seborrhea. We notice with the dermatoscope a follicular capacity of 1 to 3 hairs per follicle, but thankfully no other scalp issues. Now, let's get into the treatment process. First, we clean the patient's scalp with sodium hypochlorite. And next, we diluted the exosome solution and extracted 2.5 milliliters while adjusting microneedling device to a depth of 25 millimeters and 90 revolutions per second. Using a syringe and needle, we apply the exosomes in a linear fashion dripping along the designated areas. This was followed by microneedling up to a depth of 1 millimeter. The application was done in quadrants, targeting the apex, trichon, and interparietal region and vertex. We perform micropuncture in a circular motion until slight bleeding occurs, indicating penetration into the dermal papilla cells. The patient was then advised to let the exosomes work their magic overnight. Okay, this is a treatment combination therapy treatment in 6 steps. First step, I gave him a formula of minoxidil hair lotion every night. Application of keratin of the hydrolyzed hair lotion every morning. Guarana shampoo and ricino extract 3 times per week. Simpitronite shampoo 2 times per week. Supplements of vitamins for hair, like vitamin B5, B6, B8, B9, biotin, magnesium, daily for 4 months. And 4 total sessions of exosomes over the course of 3 months. In the first follow-up, we noticed significantly reduced hair loss, and the patient mentioned seeing baby hairs beginning to grow after week 3 in the frontoparietal area. And in the month 2, we noticed hair growth and no hair loss. And remarkably, after month 3, we saw continued regrowth of hair, 90% reduction of hair loss, and 80% of hair recovery. The patient reported satisfaction with the results with no complications such as irritation, itching, erythema, or hair loss. You can see the improvement in the hairline as the hair appears darker and thicker. His scalp feels like his scalp feels normal and not oily. Cue the comparative photographs. We can see significant hair recovery, especially in the frontoparietal area, where the hair appears thicker and darker. Additionally, there is better hair quality and more density. It seems like we have almost achieved full hair recovery. In this comparative, we can see the improvement in current area. And now, let's shift our focus to the lateral and frontal views. Look at the progression improvement in the hairline, and we're successfully recovering in the treated areas. This is beautiful, right? Well, in conclusion, after just 3 months of treatment for androgenetic alopecia, our patient experienced significant progress and improvement. This confirms the safety and effectiveness of the exosome protocol developed by ExoCoBio Benev. It's a winning combination therapy that ensures patient adherence and compliance, resulting in having 20 and 80% improvement in hair recovery and an 80% reduction in hair loss. We're making significant strides in reclaiming healthy hair. Most importantly, our patient is happy with the results. Thank you for having us here.
Thank you so much, Dr. Carlos. That's a great result. So last but not least, let me invite Dr. Daniela Mendoza from Mexico as well to talk about scalp treatments with exosomes. Thank you.
I want to ask everyone to please stay, and this is the last lecture, and then we're going to have the awards, so please don't leave.
I would really like to say a very, very sincere hamsamida to everyone who is here, because I know it's late, and I hope everyone can bear with me for the last minutes. Well, I want to present a case of a 79-year-old woman who presented to the clinic because she had fell and her skin got caught on the rails of an electric escalator. She's old, so she couldn't get up, and her hair kept getting onto the rails, and this is what happened. You can observe how the muscle was exposed and the bone also. 40 years before this patient, unfortunately, she has bad karma. She had the same accident 40 years before. She had a scalpel on her, on the scalp area, and she had gotten on that time a total depth skin graft. You can observe how the skin is hyperpigmented, it is atrophic, and it is sclerotic. It is a very bad condition of the skin. So when I saw her, I cleaned the wound and we started antibiotic treatment. And as I said, she doesn't have a very good luck. So she had a gastritis. We had to change the antibiotic. And when I saw her again on day 5, I decided to start exosomes because I was preparing her for a total skin graft again. Well, this is the treatment plan. We started doing wound treatments, and I cleaned with hypochlorite solution, and I started applying topically on the skin the exosome. It depended on how much we use. We use from 0.5 to 1 milliliter, and then I put some TV, and so she relaxed, and she didn't fall again on the clinic. And after about 10 minutes, we covered with either a duoderm or an algicide patch. It depended on how I saw the skin. And the patient was instructed to cover her skin and come back in 72 hours. During the first 26 days, we saw very good results. So about the second time she went to the clinic, we decided not to perform surgery, and we would continue just with the wound treatment. She was very desperate to go back home, and so we let her go back home, but she was not able. I didn't think about giving her the treatment so she could put it by herself in her hometown. So she went without the exosomes. This is what happened when she stopped using the exosomes. Another 24 days for the skin to heal completely. This is review of what happened in the first 24 days with exosomes, and then for the next 26 days a little slower for another 26 days until the skin completely healed. You can observe here how the skin quality is better. It's less hypopigmented and hyperpigmented, and the quality is way better, as a lot of the lecturers today have noted. So wound healing is a very important part of the dermatologic practice. We have a lot of ways to clean wounds and to heal them, but sometimes it's not very easy on all the patients. Conventional treatments can be very expensive, or sometimes the flaps are not, or the patients are not good candidates either for flaps or for total skin grafts. I think, or we thought our patient was not the best candidate since she has the very bad skin before. So the patient selection is very important, and it's very good to have other types of treatment which are not that costly, which are easy to perform and are not hurtful or harmful for the patients. Our patient had a complete wound closure in the total of 50 days with the exosome regenerative complex. Especially in this patient, we were very happy to be able to use this because she had comorbidities and bad skin quality. We were happy we didn't need to do surgery because she had to take the escalator again when she went home. This was a painful treatment, and she was very, very happy and very content with the treatment. This is a summary of what happened, and I just want to show here how the first 24 days were they were going well, and when I stopped using the exosomes, it did heal, but it was almost the same time without the exosomes. So we know in the literature, we have been using in vitro and in vivo exosomes to cure wounds. And I just want to say that this wound got better because of all the ways the exosomes work. First of all, they suppress inflammation, and then we can see how, again, there's vascular proliferation, and then that we get good healing in early and late stages of the wound healing. These are some of the mechanisms that we have already seen in some of the lectures, so I will not talk about this again. So there was an accelerated closure with the exosome regenerative complex that we did not see when we stopped using it. I really want to stress this because it is not a controlled case, but it is very evident how it was going very well and very fast, and then it stopped when we did not use it. I will take into consideration what you have done that you can give the patient to take home. This case exemplifies how there are other ways to get a good result in patients, not just skin grafts. Thank you very much for your attention. And
thank you so much, Dr. Dan. It's such insightful talk and also such a beautiful case. So after hearing 5 amazing cases here, we're moving on towards the panel discussions. Because of time, I think there's some of us with thousands of questions, and there are those who, after a long day, can't wait to go partying. So we only have 10 minutes for this Q&A session. And we will open to 2 lucky person. You have to be fastest to be in front of the mic to get that question out. Anybody? Everybody's so eager to party, I guess. You just get in front of mic. But we only limit to 2 lucky person to get that question out. Okay, yes.
In your experience, how many sessions, how can we predict
sorry,
the
can
number?
you introduce yourself, your name, and then...
My name is Juliana, I'm a dermatologist from Brazil. And thank you for your classes, were amazing. And I would like to know how can we predict the number of the sections for Asian skin, for example?
Are you directing to any person or are you anybody? Anybody?
We ask the public to please keep quiet.
Okay.
Well, actually, from my very short experience with exosomes, and remember we are working using RAES stem cells exosomes, we found that the more you apply, the better and faster results you get. So we recommended twice or even 3 times a day application.
But after treatment, like after microneedling?
After microneedling, I usually apply 3 layers, one after another. And then if there is something left in the vial, I'll give it to the patient. But usually it's enough for just next day. And usually they purchase another vial to use for next days, or they would come to the office. But we did some trials with applying just 1 layer and with the smaller frequency, then the effect was not as spectacular as with more layers and higher frequency.
I think from the study in the journal Cosmetic Dermatology this year, they show that the microneedling in combination with the adipocyte-derived stem cell exosome for 3 sessions with 3 weeks apart, with the last follow-up at 6 weeks showed its significance. So we can refer to that at least 3 sessions.
Thank you. Yes. Hi, this is Dr. Serapi from Saudi Arabia. My question is especially for Dr. Lim regarding, do you think exosome can cause cancer? Does exosome stimulate cancer cells?
Me or you?
Oh, yeah, you.
So the question is, can cancer cells exosome cause cancer?
Yeah.
That's a very good question. We don't know. So there are lots of studies that suggest it. But if you look at the methodology, it's not so robust. So I think at this moment, the jury is still out whether tumor exosomes can cause tumor.
So it's possible.
I guess nobody really works on that, right?
Jan has something to say.
All right. Okay. Yeah.
Okay. So the question is really whether an exosome can change a cell to become a cancer cell.
Yes.
There's no such evidence, as far as I know. Now, there is evidence to suggest that cancer exosomes, melanoma exosomes, potentially, and that data is not so robust either, potentially can help the metastasis of... you don't believe that? No, I said, no, I agree with her actually, but there's some paper out there that suggests that.
That study is flawed, so I'm sorry, I don't agree with it.
But I said yes, I agree with you, but there's data published that suggests that. I didn't say it does. Big difference. So I would argue no.
Okay, thank you for the question. All right, so because of time, maybe I will get one more. Last one, okay, one last one. You're gonna fight for it, three of you.
She said it's a cancer-related question.
The same one? Yeah, it's related. So it's related to the previous question. So if the patient has already previous history of cancer, are we allowed to treat it with exosome? They already had a previous history of cancer. There is any protocol? Should we have to ask every patient if before to start to treat it with exosome if they have a history of cancer or malignancy?
Anyone want to take that?
I'm not sure if this is, I think that would have to go through clinical trials, and then they will have to look at contraindications for the use of exosomes. So I don't.
Yeah, I'm thinking. Okay.
No, I don't agree with you. I see no reason if a patient. I mean, of course, you should know your patient's history.
Sure.
You have to, but putting topical exosomes on the face has no relevance to breast cancer. None.
But we saw today also one case that was treated in the breast area after breast cancer with a topical exosome. Is it allowed? Because she has already the cancer in the breast.
So what is allowed or not in your jurisdiction, I have no idea.
I mean,
I have no idea.
Is it
But I mean, the reason is to treat this radiation injury, right?
Yeah.
That makes sense.
It's radiation or scar after surgery, mastectomy, because as I remember yesterday, it was also one case, it was treated with exosome after post-radiation, post-surgery, post-mastectomy. So is it allowed to do it?
So I'll explain this a bit further. Is that okay?
Yeah.
Right. So to really transform a cell to become cancer, you need DNA. You need the DNA to transfer it into the cell and go into the nucleus and make the nucleus a cancer nucleus. I don't think there's any evidence that this really happens with exosomes. And with these type of exosomes, the amount of DNA is really extremely low. So the material is processed in such a way that the DNA is almost zero, at levels lower than what FDA would allow. So
thank you. So yeah, that's this kind of questions is always to do or not to do. I think in the end of the day, as physicians, you have to take to consider risk and benefit. For instance, if it's clearance, you know that the risk is so low, probably you will, but it's it's in the end your decision. So basically, I think because of time.
Excuse me.
Yes.
Yeah, I want to ask my question. They postponed me twice. I'm not sitting.
Okay. Okay. All right. So why don't this?
I want everyone to hear my question.
Right. Because you can always ask the question at the party, but I'll let you get the question. Okay, go ahead.
Okay, so my name is Saif. I am the distributor in the United Arab Emirates. As in the industry, every 5 minutes a new company comes with a new exosome. So every company with a new exosome claims that they have the best source. Bone marrow is better than fat cells. Umbilical cord is better than bone marrow. Human placenta is better than X and Y. We want to have a final answer. When it comes to multiple sources, which one is the best as far as we are concerned? Skin and hair.
So you want to ask the mother of exosomes?
Of course.
Of
my friend.
Okay.
I don't think there is a good answer because it depends on the manufacturer. So a good manufacturer, even using supposedly poor quality MSC like bone marrow and then somebody has something more superior. If this guy is good in the manufacturing, he can produce better exosomes than the next person. But the question is, what is better? Actually, we don't know the answer. Surprisingly, we don't know the answer. So does 10 billion cells, 10 billion exosomes better than 1 million exosomes? Maybe, maybe not. Because if you look at the numbers, it's like currency. I just told somebody. Is $1 better than one, I don't know what, Zimbabwe, $1. Both are $1, so which is better? So the particular numbers are just that. Unfortunately, we don't have an exchange rate. We don't know what is the exchange rate. So I think you will have to stick to people with good reputation, good track record. Sorry.
All right, I hope that answers your question. So we're gonna end this Q&A sessions and we're gonna move on. Any more questions you can ask during the party? Okay, thank you very much. Thank you, panelists. Moving forward.
Thank you everyone for staying so late. We're gonna go with the ExoCoBio award, so please deal with us 10 more minutes. All right, everyone. This is the most important part of the night. We're gonna have the ExoCoBio awards. So, and then we're gonna go party upstairs. So please, 10 more minutes. So we're gonna ask our CEO, Mr. Pyong, to get on the stage for the awards. So the first awards of the night are for the distributors. And then we're gonna have the awards for the speakers, and lastly, the before and after awards, the before and after pictures. All right, so we're gonna start with the distributors awards. This is done alphabetically by the name of the country. So we're gonna start first with LG Chemical from South Korea. So please a big round of applause. LG Chemicals from South Korea, and then Ven Intelligent Aesthetics from the Philippines. A big round of applause for them, please. And then Inmeris from Poland. Please get on the stage. And then Aesthetics by Ampex from Thailand. We're gonna have Aesthetics Middle East from the United Arab Emirates. Woo! Then Benev company from the US. And lastly, Saisi Aesthetics from Vietnam. So a big round of applause for all of them, please. All right, a big round of applause, please, for all of our distributors. We also want to say thank you to all of the other distributors who are not called today. And Benev from the USA. So we're going to take a quick picture. Thank you to all of them, and thank you for the distributors who are not awarded tonight, but anyway, you came to Korea. We appreciate all of you. Thank you very much for joining us tonight. Okay, now we're gonna go to the second part, which is the Speakers Awards. So to all of the speakers, please get ready. So I'm gonna call one by one. Dr. Jan Latval from Sweden. A big round of applause, please, for Dr. Jan Latval. Then Dr. Michael Gold from the USA. Dr. Renato Salz, also from the USA. Dr. Íñigo de Felipe from Spain. A big round of applause for all of them, please. That's all. Oh, he was there already. Okay, he's there. Dr. Rosario Salud. Okay, Dr. Rosario Salud Abaya. A big round of applause for her, please. Now, Dr. Julia Tabarani O'Shea from Ireland. Please, Dr. Julia, welcome. Now, Dr. Ali Alambri for the Saudi Arabia. Yes, Dr. Ali Alambri from Saudi Arabia. Please, a big round of applause for Dr. Alambri. And finally, but not least, Dr. Victoria Bello from the Philippines. Yes, Dr. Victoria Bello from the Philippines. So please, a big round of applause for all of them, the speakers tonight. So we're gonna have a picture all together, all the speakers. Dr. Fernando Macedo from Brazil. I think, okay. Dr. Sai Kyang Lim from Singapore. Dr. Tom Siri from the US. From Dr. Richard Jim from the US. Dr. Dot Schlesinger from the US as well. Dr. Lim Ding Sung from Malaysia. And Dr. Arjun Jasdani from Canada. Please, a big round of applause for Dr. Yazdani. And then we're going to have a picture with all the speakers. Oh, Doctor, wait, we have a lot of speakers tonight. I know it's been a long night, so please be here with us. And then again, Dr. Lim Ding Sung, please. Big round of applause for him. So let's take one big picture, please, with the speakers. And then one more round of applause for them, please. And look at the cameras. Thank you very much to all of you. And now, we're gonna go with the last part of the night, the before and after awards. The before and after pictures awards. We're gonna have time at the end for pictures, so please don't worry. We're gonna have time at the end for the pictures, so please, we still have one more, 10 more winners. Dr. Fernando Macedo is here. Please, Dr. Fernando Macedo. You want to come this way? Dr. Fernando Macedo from Brazil. Please, a big round of applause for him. And then we're going to ask the rest to please sit down because we have one more activity. So please sit down if you can help us with that. Lugan. Yeah, but then. So, all right, so we're gonna go with the before and after awards. We're gonna start with the third place, Dr. Ana Cecilia Amador Ruiz from Mexico. Please. Congratulations to Dr. Ana Cecilia Amador. Then we're gonna and then Dr. Jonah Luis Macarach Jimenez from the Philippines. There is Dr. Jonah. Congratulations. Yeah. Congratulations. Remember that for 2 months we were collecting cases from all around the world. So these are the best top cases that we received. Big round of applause for Dr. Yona, please. And then Dr. Jacqueline Luna, also from the Philippines. Oh, okay. Okay, she's actually Dr. Luna. She was getting the plaque for the other doctor. So please, one more round of applause for her and one more picture. Woo! Then Dr. Guada Capiz, also from the Philippines. She's coming there, please. One big round of applause for her. Congratulations, doctor. And we're gonna have Dr. Suramis Estrada Blanco from Spain. She's not here tonight, so Mr. Marcos will receive the prize for her. So welcome. Please, Marcos. Mr. Marcos is the distributor of Belgium from Spain, so please a big round of applause for him. Now Dr. Jennifer Levin from the USA. A big round of applause for her, please. Thank you very much for Dr. Jennifer Levin. Woo! Now, Dr. Carlos Alberto Garcia Meraz from Mexico. Thank you very much. Doctor, congratulations. There we go. Congratulations, Dr. Carlos. Nice pictures there. Felicitaciones. And the Dr. Daniela Gutierrez Mendoza, also from Mexico. Thank you very much. And thank you to all of you for staying until late at night. Yes, very nice. So a picture with all of them on our CEO. Right, a group picture. In Korea, it's always picture. All right, smile. Kimchi for the picture. Kimchi. No, they don't want to say it, but it's okay. All right. Again, congratulations to all of you. And now we're gonna announce the second place. Second place. All right, the second place, Dr. Suparu from Thailand. Woo, Dr. Suparu. Congratulations to Dr. Suparu. It's $5,000 for Dr. Suparu. And then Dr. Lidia Majewska from Poland. Big applause for her, please. Congratulations, Dr. Lidia. Woo! All right, she's holding it like a trophy. Amazing. Congratulations again for them. Please, one big round of applause for them. Amazing cases tonight. Amazing. Thank you very much. And now, the most expected time of the night. We're gonna announce the first place. All right, are you ready for this? All right. I would like to I will say thank you to all of you also for staying until late at night. After we're gonna go party. After the prize, we're gonna have some pictures. So please, 10 more minutes. All right. So the first place goes to Dr. Jose Ramon Garcia from Mexico. Woo! Congratulations. Congratulations, Dr. José Ramón García. Felicitaciones en español. Oh, very nice. Very nice. Kimchi, you have to say kimchi for the picture. That's $10,000 in cash that he's gonna spend tonight in the club. Okay. That's Emmanuel in the picture. Very nice. Congratulations. Felicitaciones. we're gonna have the closing words from our CEO, Mr. Byung. Then we're gonna have pictures and then party. So wait, please. We're gonna have one more closing remarks by Mr. Byung, and then we'll take the pictures.
All right, thank you so much for all day, for waiting for this moment, for the party, right? Everybody's ready for some drinks? Champagne?
Yes.
Cocktails? Yes. Yeah, 30th floor and the latitude. I'd like to thank you all for coming out here and enjoy the lecture and celebrate the success of others and paying attention to what could definitely change the entire industry. And I'd like to thank Byung and again, ExoCoBio staff to putting it all together. Again, do not miss out today's party. It's gonna be the most spectacular view you're gonna see in the night view of Seoul. Great music, great drinks, and we have a virgin drinks as well too. And I'd like to invite as many of you as possible to come forward so we can take a group picture.
Yes, so please, all of you, please come forward. We're gonna take a group picture, so let's quickly gather here. All of you, please, all of you. Please, all of you, please, if you wanna come to the stage, we're gonna have some pictures all together. Yeah. It's just, please, everyone, please come to the stage. We're gonna have a group picture. All right, Doctor.
Thank you very much.
Obrigado.
Muito obrigado,
We're
gonna break everything. We're gonna see you. We're gonna see you. Nice thing.
Yeah. Please, everyone, come upstage. We'll come to the stage for the picture. We're gonna have a group picture, so we want all of you to be there. Otherwise, it's gonna take 1 hour if we split it by teams. So let's do it at one big group picture. And then upstairs, there's a nice bar, so we can take more pictures there. Okay, is everyone ready? You have to say kimchi, right? All right, are you ready? Kimchi, kimchi, kimchi. Kimchi, okay, one more. One more time. All right, kimchi, kimchi and the heart. And then fighting, fighting. Okay, you want to say Diego? All right, thank you very much. Thank you to all of you. So, everything is already set up upstairs. Latitude bar, 32 floor. together. Yes. So the party is gonna start. It's starting right now. 9:30 is already set up in the 32 floor Latitude bar. So please, if you wanna get changed, there's no time. So just go party like you are right now. There's some cocktails, a lot of drinks. Everyone, I want to invite you to please get ready for tonight because it's gonna be endless. 6 a.m., more 6 hours of cocktails and party and fun. Saif.
Yeah, with Diego. Yalla. Viva Colombia.
Viva. Nice. Congratulations. Looking very good. Yeah, but it's nice. I'm very happy to see you. I'm very proud actually.
All I think.
I'm proud, like, wow, it's our thing, it's our hard.
Received from Gillian and when we reached to
الحمد لله سيف خلينا نتصور فيها قول اعلى محقق في العالم.
All right, everyone, please let's slowly get ready for the Latitude bar in floor 32. The cocktails are waiting for you. The view is amazing there, so you can take more pictures upstairs.
Recording stopped.
Okay, cool.